Okay, so the SACATA members are all present. So if we can finish our conversations in the room, I'm going to introduce the next topic, which is the NAMS pipeline future directions. And I'd like to introduce Dr. Gadbois to talk about that. Thank you.
Looks good. Thanks very much. It's a pleasure to be able to talk to you all this afternoon. I am here to give an introduction to the NIH, the current NIH vision of what we refer to as New Approach Methods, or NAMS. Today, I will be giving an overview of recommendations from the NIH's Advisory Committee to the Director, the ACD. These recommendations have been accepted by the NIH Director, so I'll be talking about those with an emphasis on what my office thought was of most interest to this group, and then I will be turning to my colleague in the NIH Office of Strategic Coordination, talking about a new NIH program that is specifically designed to advance the development and the use of these new approach methods. Next slide. Thank you. So the impetus for the work that I'm talking about today came about, at least in part, due to a growing recognition of the value and the increasing sophistication of model systems being used in biomedical research. Systems that are not animal models and that enable researchers to ask questions using human systems quite quickly and at a larger scale than can be done with animal models. So this slide shows a few examples of human cell-based systems that are exciting and show ways to do experiments that would have been difficult in the past. So back in 2022, NIH decided that this was a good time to look more closely at the state of the science using new approach methods and consider how NIH can keep moving fields forward. What are the challenges and opportunities and the investments needed? And let me turn off my phone, which is giving an alarm, reminding me that I'm supposed to come over here and do this talk. All right. Thank you. Next slide. All right. So at the end of 2022, the NIH acting director, Lawrence Tabak, convened a new working group of the Director's Advisory Committee. You will see a few familiar faces here. This working group had broad membership in terms of expertise, fields represented, folks in academia, in industry, several ex officio members from our components of NIH and other federal agencies that are interested in this area. And the charge to the working group was to consider the development and the use of new approach methods, including identifying types of alternative methods and assessing their strengths and weaknesses with a focus on studying human biology. Thinking about the kinds of research that they are most appropriate or beneficial for, and then articulating what could be NIH priorities for advancing our understanding of biological processes and augmenting the tools and the capabilities for researchers to complement or potentially replace traditional models, including animal models. So this working group thought about this area over the course of a year. There was a request for information issued. There were 2 workshops held. So they gathered a lot of information about what was going on and what people were thinking about going forward. Next slide, please. So the working group developed this scope for new approach methods with 3 modes: in chemico, in vitro, and in silico. Some examples listed on the slides of what they saw falling into those 3 areas. So the working group and NIH hope to see some of these also used together in the future in what we call combinatorial NAMs. And as I'm sure many of you would know, that they are useful for a range of research, very basic research, research understanding human pathology, human physiology, and then finally looking at products, practices through use in toxicological testing, drug screening. So we see a variety of approaches using some very different starting technologies, but all of which have the common feature of being new methods for new tools for investigators. Next slide, please. So this next slide is an example that the working group used as an illustration in their presentation to the full advisory committee, to the director. One example with studying metastasis of cancer cells and the challenges there, and how the working group envisioned new methods being used to give a different approach to studying what happens with cancer metastasis. Starting with the patient and developing organ or tissue chips from that patient using various technologies, imaging, omics, computational modeling to study those tissue chips, and then benchmarking that data to other available data, clinical data, data from traditional animal models, all of which we hope will ultimately benefit research participants and potentially patients. Next slide, please. So the working group then developed this overall view or vision for new approach methods. So it's both the working group vision and call it the NIH vision as well, since the NIH director embraced these recommendations. So at the top, starting with what the working group focused on was combinatorial NAMs, combined or integrated technologies, which they thought were really key to maximizing the utility of these approaches. Developing, using those NAMs within multidisciplinary research teams, since so many of those methods need people coming from different disciplines who would benefit from training, especially with some of these more sophisticated methods, have access to high quality data sets, but then be also charged with passing on their knowledge, their methods as part of an ongoing productive cycle. Next slide, please. So the recommendations from the working group fell into 7 thematic areas that are all listed here. And many of these approaches are bread and butter for NIH in different areas of research. Here they are cast in terms of how the development, use, further education with new approach methods can benefit, but they're also relevant for the broader biomedical research ecosystem, and there are likely groups outside of NIH who, if they take on these activities, can strengthen the whole field. Here at NIH, we're thinking about what we can do, but there are certainly things outside of the NIH supported community that folks who could carry out some of these activities. So I thought I would go through these thematic areas and just highlight some of the recommendations under them that my office thought would be of most interest to you all. So I'm not going through everything, and the report and the presentation of the ACD are all on the NIH ACD website from the December 2023 meeting. So welcome to look there for more detail. But I'll just walk through the 7 areas with what we thought you might be most interested in, and then I will be turning over to my colleague, Margaret Hochinska, who will show you a very concrete example of what NIH is doing to work towards some of these goals. Next slide, please. All right, thanks. So recommendation 1 deals with prioritizing development and use of the combinatorial NAM, so integrating or combining some of these technologies. And in this case, the working group thought it was very important to be developing benchmarks, standards for these methods, and support research comparing them to what else is out there, the relevant animal models, other approaches, so we can best validate the specific NAMs being developed and used. Next slide, please. Okay, this slide, there's an asterisk here at the bottom, and you'll see a few more of those. Those are things we wanted to point out in particular to this community if you have ideas on how to advance these goals. So recommendation 2 is about establishing resources, infrastructure, collaboration to promote high quality and interoperable data sets. So this includes thinking about places, repositories where this data can go, and then creating alliances to help manage, share, let folks know about data from high quality NAM. So one thing in particular, if you have thoughts about ways to share unpublished data, that's always something that we think is important, but not always obvious how to do that. Next slide, please. All right, recommendation 3 is about effective dissemination. So mechanisms to support testing, validation, qualification, and then again to have sources, repositories for this information so that people know these are available as tools to consider using their research. Next slide, please. Recommendation 4 is about investing in training across the pipeline in academia, creating collaborations between researchers and clinicians, fostering entrepreneurship training, and then promoting awareness more generally of new approach methods through things like course modules, workshops. Again, there are things here where if they resonate with you all, we'd be interested in hearing what ideas you might have about ways to do this. Next slide. So recommendation 5 is about facilitating multidisciplinary teams. Again, since many of these draw on different areas of expertise, also highlighted some regulatory policy issues that may arise here, since these are new things for eventual use in the regulatory environment. Folks will need to think about how to make that happen so we can maximize the value of these methods. Next slide, please. Recommendation 6 is about promoting social responsibility. NIH spends a lot of time thinking about how to third bullet in particular, how to openly share technology, data, and try and best represent different communities, diversity of patient population, and be thoughtful in the origins of that information. Then a second point here is about strengthening interagency partnerships to advance NAMs in a way we think is appropriate. All right, recommendation 7, last one. There's a lot here. So the working group wanted to see mechanisms for disseminating resources as they are developed, the protocol, thinking about clearinghouses, knowledge bases, and then promoting ways in which, through consortia or other venues, to share things that folks have learned in developing NAMs as part of their research. All right. So next slide, please. Okay, I'm going to end here and just let you know that my office, Deepa Kipsey, the Division of Program Coordination, Planning, and Strategic Initiatives in the Office of the Director, is leading implementation of these recommendations, which has involved contacting all of our institute centers and offices, making sure they are aware of these recommendations, asking what they are doing in this area, trying to stimulate further thinking and collaboration and pushing this forward. So I think this is something that we will be talking about in terms of these recommendations for quite some time. But I hope you appreciate that we have also a very new exciting program that is launching, that is focused on this area and is a way to bring about many of these recommendations in a high-profile NIH program. So I'm going to stop here. I don't know if we want to take any questions now or if we'll go until later.
Yeah, we're going to open it up for clarifying questions only at this time. Please hold your discussion questions until the end of this session. I do not have any hands up on the Zoom at the moment.
Thanks. Thanks very much, Ellen, for the overview. And I'll just add one additional sort of clarifying point that first, a big thank you for stepping in because Ellen took this over from another member of Deepa Kipsey who was leading this. And I just wanted to note that we have engaged over the last year with Deepa Kipsey and with the office that is leading these implementations to identify recommendations and sub-recommendations where ICFAM is actually already engaged in those spaces to implement and operationalize some of those recommendations. And so I think there is an awareness that there is activities ongoing already across the federal government to address these recommendations. But we're, hoping obviously in the discussion today to talk about what more can we do to help.
Yeah, thank you, Nicole. And I should have recognized that ICFAM has so much already going on. I mean, these recommendations are in some cases talk about things that are active in many NIH institutes and centers and programs. I think part of the value of the report is that it stimulates people more broadly to think about these things, but we already have a great research base.
Okay. Yeah, we don't have any more clarifying questions at this time. So we're going to move on to the next topic. Thank you so much. We are going to have the complementary program overview from Margaret Oczynska. Yes, that's fine.
Good afternoon. Hopefully you guys can hear me. Looks like it's working. Okay. Thank you so much for the opportunity to present an overview and update on the complementary program, which stands for Complement Animal Research and Experimentation. And my name is Margaret. I'm the program leader from the Reengineering the Research Enterprise, a very apt moniker for this, from the Office of Strategic Coordination at NIH. Next slide. First, before I go into the details of the program and the strategic planning process that was involved, I wanted to provide a bit more background on the Common Fund, because some of you may not be as familiar with the Common Fund process. So essentially, this was built from a 2004 NIH roadmap that was launched. And in 2006, Congress unanimously reauthorized the NIH and established the Division of Program Coordination, Planning, and Strategic Initiatives, DPCIPSI, within the Office of the Director and the NIH Common Fund to provide a dedicated source of funding to enable trans-NIH research. So before, the model was for NIH institutes and centers to contribute funds to a pot for trans-NIH activities. Here we have a set-aside fund for trans-NIH research. So in terms of the Common Fund mission, it's to move the NIH mission forward faster by supporting bold scientific programs that catalyze discovery across all biomedical and behavioral research to advance areas of biomedical and behavioral research important to the missions of multiple NIH institutes and centers, and to spur subsequent biomedical advances that otherwise would not be possible without an initial strategic investment. And in terms of the feature of Common Fund programs, because there's a long selection process for ideas to be developed for the Common Fund, the features of these proposals need to make substantial investments in time-limited, goal-driven programs that significantly change the trajectory of biomedical research by accelerating emerging science, or removing research roadblocks, or enhancing the research workforce, or supporting high-risk, high-reward science. And how this is done is through a Common Fund science and management through a collaborative process. So scientists from diverse disciplines are asked to provide input as we plan new programs. Researchers are asked to participate in interdisciplinary consortia to tackle shared goals. And leadership and staff from multiple institutes and centers and offices of the director and across NIH work together to design the funding opportunities and oversee the project. And so today I'm going to tell you a bit more about one of those programs that has been discussed and mentioned earlier. That's the complementary program. So the purpose of this program is to catalyze the development, standardization, validation, and use of human-based new approach methodologies, NAMs, that will transform the way we do basic translational and clinical sciences. So it has several overarching goals. One is to better model and understand human health and disease across outcomes across diverse populations. 2, to develop NAMs that provide insight into specific biological processes or disease states. 3 is to validate mature NAMs to support regulatory use and standardization. And 4 is to complement traditional models and make biomedical research more effective and efficient. And just to illustrate the point about how trans-NIH this activity was, this is sort of illustrates the breadth of expertise across NIH. This includes more than 23 institutes, centers, and offices. And as you can see, it's co-led by Joni Rutter from NCATS and Rick Wojcik from NIHS. And then we have working group coordinators from both institutes. And then we have more than 60 members of the working group, including, you might recognize that name, Nicole Klein-Steuer, who is the strategic visionary for the strategic planning process for this program. And so again, to illustrate the point, this is trans-NIH, and as you will learn, it's also trans-agency, and we certainly hope to partner with multiple stakeholders in this space and to leverage the exam community as well. So in terms of the timeline, so Ellen mentioned a bit more about the NIH Advisory Committee, the Director Working Group. So when our program concept was approved for strategic development, this happened last summer. The Common Fund selected this concept proposal and convened the working group. That was last summer in June. This was around the same time that the ACD working group presented their preliminary findings. So there was sort of a parallel track for both activities. We were asked to do the strategic planning that's listed on the left, while additional activities were ongoing, and we had a exchange of information between the two groups. And so here we really wanted to inform the strategic planning through what was also gathered from the ACD working group findings. So for our part of the strategic planning process, we included a deep landscape analysis to look at data infrastructure needs, assess current use of NAMs, and what are the gaps and challenges. We also held listening sessions and roundtables to engage the scientific community to gather input. We also had an interagency workshop and an ideation design prize. And I will tell you more about those in the next couple of slides. Suffice it to say, the Council of Councils approved the concept in January of this year. And so we've been undergoing a very detailed implementation planning process with our working group members. So I will tell you more about what has been developed thus far. So essentially, the general program goals include complex in vitro models emulating population diversity, also in silico multi-scale systems simulating healthy diseased individuals, also in chemical cell-free systems capturing dynamic changes, and as well as combinatorial NAMs and integrated fair data sets for all NAMs. And along these lines, we definitely want to build off of the successes across NIH. So there were multiple advances made over the course of several decades through NIH and other agencies. And here's a highlight of the NAMS activities across NIH, including digital twin models, in silico models, complex in vitro systems, and chemical screening. And really complementary aims to integrate those solutions into a comprehensive new approach methodology approach that will learn from these various different paths. And so really we wanted to provide more information to you in particular about our strategic planning process. So as I mentioned, we had the listening sessions that included academia, industry, NGOs, government, and international partners. We also had an interagency retreat October 19th and 20th last year. And so that included NIH, FDA, EPA, NSF, ARPA-H, BARDA, VA, DARPA, NIST, NASA, CPSC, and ICFAM members. So some of you have participated. Thank you so much for attending that interagency meeting. And the summary from that meeting is available on our complimentary website. We also identified some priority areas and scientific needs, such as chronicity, neuroscience, personalized health, cross-disease pathogenesis, and population diversity. We also identified operational needs, integration, coordination, and collaboration through shared data infrastructure and standardized frameworks, validation, and training. And at the bottom of the slide, we also have links for the Ideation Challenge Prize, as well as the landscape analysis summary. So those are also available. So the winners have been announced. So those are accessible on our website in terms of information of the type of solutions that were submitted. And here's more about that prize competition. So really we solicited, we used a different approach to crowdsource potential integrated NAM solutions from the community as part of the strategic planning process. So rather than waiting for the program to be launched and then do a challenge initiative, we decided to solicit ideas from the community for an ideation challenge prize. And so you can also access and view the winners. So we selected 20 winners from the submissions this past spring. And so the solutions that came in, we also used to inform our strategic planning process to identify additional gap areas or potential areas of need for future funding opportunities. And here's just a highlight of 3 of those submissions that are part of the 20 winners. So one is the Click Chemistry Magic for Drug Screening, which is a novel chemistry name for high throughput drug screening, tissue engineering, and precision medicine. And the team was Delaware Click Chemistry. And through these slides, you can click on that, pun intended, to see more details about this particular solution. We also have DevTox, Developmental Toxicity Predictor, comprehensive approach for developmental toxicity prediction to enhance maternal drug safety. And the team was Predictive LLC. And then the last one is the Pulse of the Future, AI Human MEHCs, integrated solutions to analyze multi-scale engineered heart constructs. and model human cardiovascular health and disease. And the team was Pulse of the Future, AI Human, and EHC. So you can sort of see the breadth and across the various disease areas and applications that could be possible through a complementary program, and what activities are being done and what ideas could potentially be developed further. And so here, without further ado, just wanted to highlight what are the current program components of the complementary program. One component is the technology development centers to stimulate the development of NAMs to fill in areas of greatest need. And that's really informed by the high priority areas that were identified through our strategic planning process. And that's with emphasis on increased biological complexity and throughput, innovative combinatorial approaches, and data sharing. The next component or pillar of the program are the NAMS Data Hub and Coordinating Center. We have come up with an acronym, NDHCC, to create integrated data structures, including standards for monocodability, which is very important in developing NAMs, as you know, and to improve fairness and of NAMs relevant data and create searchable NAMs repository. And the third pillar that you'll hear a bit more about in the next presentation is the Validation and Qualification Network, or VQN, for NAMs adoption and implementation, to establish common data elements and standardized reporting to apply validation and qualification frameworks and accelerate deployment and regulatory implementation of NAMs. So those are the three key pillars, but underneath all of these pillars, we want to include community engagement and training to promote the development of an inclusive, diverse biomedical research workforce with the skills to build and use the NAMs that are developed, to have community engagement, to have data sharing, to have information sharing across the scientific community, but also communities at large, to understand how there's a paradigm shift from reducing, refining, replacing animal models to identifying ways to use new approach methodologies to help in that process. And of course, we want to include societal and ethical considerations for this as well. And so essentially, this is for those that are visual, we have just a visual representation of the technology development centers and how they're placed within the framework of the NAMS Data Hub and Coordinating Center. So we anticipate finding 5 to 6 centers, with perhaps additional centers co-funded by additional federal agencies. And the NAMS Data Hub and Coordinating Center will, of course, serve as both a coordinating center and the data hub to have this interface with the centers, as well as this other component, which is the validation and qualification NAM for NAMS adoption and implementation. That's going to be an Uber convener for other agencies, for exam members, for regulatory partners, for industry partners, nonprofits, and others. And I'll tell you a little bit more about it as a preview in the next couple of slides. So the first announcement I wanted to just share that's already been public or earlier, made public earlier this month, is we have this notice of intent to publish a funding opportunity for the technology development centers. So the estimated release date of this NOFO, it will be end of October, so around October 18th or a little bit later, but within October. And you can also see additional information about this activity on our website. And we will be, once this is published, if you want to sign up for the listserv and also share broadly with your community, we will make an announcement for when that funding opportunity is available. And then the last couple of slides, I just wanted to highlight this piece where we're exploring the potential public-private partnership through a validation and qualification network for adoption and implementation of NAMs. And really, this way we're exploring ways to bring industry, NGOs, nonprofits into the network by leveraging existing FNIH public-private partnership infrastructure, as well as to be able to obtain pre-competitive data sharing and potentially supporting validation activities across labs and locations for specific use cases for implementation, and to support community outreach and training. So again, one of the key pillars and underlying substrates for this, and to provide a fluid funnel for potential solutions that could be developed through the technology development centers. And we are exploring including additional federal partners in this activity and to synergize and coordinate with other global activities on NAMs. So here is just a broad overview of this 3-phase approach that we are exploring that would include a 1-year design phase to plan and develop this network with the industry, NGOs, CROs, and federal partners to determine the scope of the validation efforts to develop governance criteria and selection of use cases and to refine the goals of the network. And we are certainly exploring ways to leverage the XCAM subgroup to advise on validation steps. So we want to hear from the experts on this process. And we would be holding workshops with stakeholders during the design phase to develop the implementation phase, which we envision to start when the complementary program launches a year from now. So the implementation phase would include implementing validation efforts with the partners, select the use cases, and fund at least 8 use case validation studies, and also include sharing data infrastructure within the data hub and the training components, and to develop long-term sustainability plans. And with the Common Fund, typically it's 5 to 10 years in length, so we anticipate having another phase, which would transition to implementation phase 2, where there will be additional activities to apply reporting standards to new NAMs and tech dev centers, and to continue validation and qualification support during that phase of the program. So I think this is just a summary of this. So here are the 3 pillars. This is the flow from technology development, centralized data resources, and validation and qualification components, where we really feel that we want to build a fluid conduit from stakeholder engagement to implementation, and relying on training and dissemination as a substrate for this activity. So sort of bridging toxicology experts with biomedical science to really, so that's why we have the conduit, and that's why we have substrates, and we have all the reference terms that would be relevant to a multi-stakeholder project. And so really complementary aims to innovate understanding of human health and disease, to integrate innovative NAMs with AI and fair data ecosystems, and to coordinate with institutes, centers, agencies, and public-private partnerships to transform the way we do basic translational and clinical sciences by leveraging the full scientific toolbox. So with that, I just wanted to thank you for the opportunity to share this with you. And if you'd like to learn more, there's the link there. It's very dark blue, but it takes you to the complementary website. Thank you. Happy to take questions.
Wonderful. So at this time, if there are any clarification questions, it looks like there are. Sally, do you want to go ahead?
Yeah, excellent job. You guys are doing such amazing work, and thank you for all of that. One clarification, you mentioned that you said it really fast, that you look at this as a way, like a replacement for the 3, or a different way, like moving away from the 3 Rs towards NAMs. Can you say that again?
Sorry,
slower.
I was speaking a bit too quickly. Sorry about that.
No.
So essentially, the goal of the complementary program is to develop integrated solutions to refine, reduce, and potentially replace animal models. So it's definitely within the 3R mission space. And what we want to, it was very intentional to call it complementary, complementing animal research and experimentation, because we don't want to necessarily replace all models. We want to complement existing models and, help the development process for new approach methodologies.
Dr. Yusio?
Hi, thank you so much for the talk. I think this is a great topic. Can you just clarify a little bit how you relate to NCATs? Like, is it an extension of NCATs, or is it sort of complementary?
So that's a great question. Another reason we're called complementary. But so the Common Fund program model is typically led by multiple institutes and centers. So if it's within the mission space of multiple institutes, in this case, the concept was developed between NCATS and NIEHS in partnership. So that's why Joni Rutter and Rick Wojcik are the co-chairs for this program. Of course, there's interest across NIH. But and then the 2 program coordinators, one is through NCATS and the other one is through NIEHS. So it's related to NCATS because it's within the mission, but it's to help create this complementary program, which is Trans NIH. So it definitely has membership from NCATS, and also, as I mentioned, the 23 other institutes and centers and offices. So, and that's part of the criteria to be selected for Common Fund is to make sure that the program is relevant to multiple ICs. And so definitely major interest within NCATS and their mission space. And in fact, it will be building upon the in vitro MPS space that NCATS has developed so well thus far. and as well as other institutes, but then taking it farther to integrate. So that one slide that I shared where, many activities are ongoing across NIH, but this program will aim to integrate those solutions to solicit applications that will provide multi-stakeholder engagement, but also different areas of science. So, bioengineers with toxicologists, with biomedical scientists. So we're asking for a lot, but we think that the field may be ready based on their recommendations to take this next step.
Okay, I don't think that there's any more. Oh, one more. Dr. Elliott.
Oh, great. Yeah, no, great talk. And I asked this question. NCATS has been doing in vitro assays for a long time. How do you reframe the problem appropriately in that complementary thing to add value to the tremendous amount of work that in vitro assays have gone through at NIH? What different and new things does this add to the in vitro assay portfolio that the NTP and everybody's been involved in? What different thinking is being brought to it, I guess, in this program?
Thank you so much for that question. So that's actually a great question because we really aim for this program. So when I mentioned integration, it means that we're looking for solutions that include at least two of the key components. So we mentioned in vitro, in silico, and in chemical approaches. So we would like the future potential solutions for the technology development centers to include at least two of those approaches. So really the aim is to get the in vitro model developers to talk to the in silico model developers, to potentially talk to the in chemical model developers, and to create a comprehensive potential solution. So the idea is to perhaps in the future, aspirationally, that can have some personalized avatars, personalized in vitro, in chemical, and silico solutions. And this is why we did that crowdsourcing approach to the challenge prize to see what crazy ideas are out there, what ideas could potentially be feasible, can this be done? And based on that feedback we received, that could be done with funding attached to it. So that's the additional piece, that this funding will be targeted to the comprehensive proposals that include both in vitro. We will not consider just in vitro.
Great, thank you. Sorry, press the wrong thing. Okay, now we are going to move on to the next section, which is the public comments for session 2. Oh, we do. Sorry. Oh my gosh.
You're fine. Right over you. I promise I'll make it quick.
We have loads of time. We're way ahead. It's all good. Just take your time. So sorry about that. I'm like getting ahead of myself. Okay, so we're going to hear from Stacey Adams on the NIH status update. Thank you.
Thank you for that. So thanks everybody for the invite to be here. I guess you're probably wondering why yet a third talk kind of on this stack of programs. So for those who may not know, the Foundation for the National Institutes of Health is the congressionally mandated nonprofit set up to help the NIH do public-private partnerships. For those who work in the federal government, many of the federal scientific agencies have something that looks like us, the CDC Foundation, Reagan-Udall for FDA. So we've been in existence over 25 years. Our whole process is to build bridges to breakthroughs. So we aren't recreating the wheel, we aren't creating anything novel. Our whole goal is to link everything going well in the public sector with all the good things in the private sector so both can go forward even better. Because while we understand that science has the power to do a lot of good things, there isn't a single organization that's going to do it alone, and that includes the NIH. So as much as we love the NIH. And just I'm giving a little bit of background on FNIH because I know that even folks within the NIH don't often know of our existence or what it is exactly that we do. So if this is a repeat for people, I do apologize for that. But we do, as I said, PPPs for NIH. We work to partner with world-class organizations. This is both our public sector colleagues, NIH being our primary focus, but we can work to link up cross-USG and even other governments outside of the US government agencies together. We do a lot of work with EMA, other places across the globe. We do a lot of work with biopharma, and that's both small and large, so large pharma companies, biotech, diagnostics. We work with just about everybody in that space. And then other foundations, so lots of work, the Bill and Melinda Gates, the Wellcome Foundation, Hemsley Trust, other places like that. So really anybody and everybody who would like to come and work with us, we try not to take that off the table and bring that to bear on things that NIH is doing. We've been quite successful in this area. As I said, we've been at this for a little over 25 years. We've got about $1.5 billion of private sector funds raised. We have 120 plus active projects, and 90 cents of every dollar that we bring in goes back out to the science. So we get a really good rating on Charity Navigator as far as if you're tracking that. We put good, the money that comes to us, we put it to good use, and we think that good use is science. And so we sort of centralize around 3 pillars. Acceleration of prevention, new therapies, diagnostics, potential cures. This is a lot of what we do in our translational science department, for which I am a vice president in that department and oversee about half of our portfolios there. We do a lot to advance global health and equitable care. This is a lot of the work that we do with the Gates Foundation in conjunction with NIAID and ICHD. And then we also power the future of science by celebrating future scientists. We award prizes, nothing to the contingent of what NIH awards as far as granting, but we do like to recognize particularly standout awards. And we have a pretty good track record, especially on our Lurie Prize. We've had a couple people go on to win Nobel, so we've gotten them a couple years ahead of their Nobel. But we just try and improve and move science forward. And so what does this look like when we talk about public-private partnerships? Well, we can do things where it's exclusively funded by public sector organizations. For anybody who didn't want to forget the last 4 years that it was the COVID pandemic, we were one of the primary facilitators for the ACTIVE program, so the Accelerating COVID Therapeutic Interventions and Vaccines. We helped to prioritize a lot of preclinical assets, and we helped to design, stand up, and prioritize the agents for the 11 master protocols that did all the therapeutic testing that essentially became the arm of Operation Warp, the therapeutic arm of Operation Warp Speed. So things that look like that, all of that was actually, like, our work was government funded because we wanted to move quickly. We didn't want to have to negotiate contracts all the way, but we brought the private sector in that case through drug donation. So all the drugs that were used in the trials were donated. We didn't pay for any of that. Pharma came to the table to help us design. All that manpower was donated. You can go all the way to the other end of the spectrum, and things like the Biomarkers Consortium, which is approaching its 20th anniversary. We put together small hypothesis driven, and by small, I mean somewhere in the ballpark of $2 to $5 million projects with anywhere from 5 to 20 partners to develop biomarkers to advance them down the regulatory development pathway. So we work strongly in conjunction with FDA, but those are almost exclusively funded by the private sector, pharma, diagnostic companies, that sort of thing. And then the middle section, which is where our sweet spot lies, which is where we kind of, government puts up half the funding, half the resources, and the private sector puts up the other half. And we act as the go-between and the coordinator for making sure that both processes are moving in the same direction and we aren't working cross purposes. And so just for some examples, and I give these and I will go through them quickly, but this is just to kind of get you thinking the art of the possible about when we talk about NAMs, how might the foundation be helpful? How have we been helpful in the past for other things that we've done? So the Accelerating Medicines Partnerships is one of our largest platform consortia. It has a number of smaller disease-focused consortiums that lie below it. It is pre-competitive. We view these as target identification and validation programs, predominantly lined up to aggregate large data sets across specific disease areas, and then integrate those together on a single platform and develop cutting-edge tools for analysis and that sort of thing. And as I said, we've been at this, we just celebrated the 10th anniversary of AMP for the first 3 that were launched, which were Alzheimer's disease, type 2 diabetes, rheumatoid arthritis, and lupus. All of the first 5 went on and converted. Alzheimer's disease continued out. Type 2 became common metabolic disease. Rheumatoid arthritis, lupus became autoimmune and immune-mediated diseases. And behind those, you can see we've subsequently launched a number of projects. Parkinson's disease, which just re-upped for Parkinson's and Parkinson's-related disease, schizophrenia. Heart failure, we just launched one in ALS. We have other ones in design. And you'll see I intentionally skipped over that one. We have something in bespoke gene therapy, which doesn't look like the rest of them. That one was actually a bit of a deviation. It's not a data aggregation. The FDA actually approached us and said, we have this idea that we could put together viral vectors, that we could make success, we could develop these things, and we could harmonize and standardize the manufacturing and testing process and get this going much quicker, and we could do this for rare diseases. So Peter Marks actually sits as our co-chair for that one. And if you saw the platform therapeutic guidance that just came out and the playbook that came out probably earlier this year, a lot of that work has emerged from the biomarker or from the AMP Bespoke Gene Therapy Consortium. So we're really lucky to have FDA sitting at the table. This is just the numbers for this. 12 projects, 915 million raised, 10 plus years with this, 36 industry partners, 16 institutes. So by the way, NAMS is already beating us on the institute count, and 43 nonprofits involved in these. The other one that I'll just briefly go over is the Biomarkers Consortium. As I said, this has been ongoing for about 20 years. The driving principles are to bridge the gaps between basic science and the practical needs for advancing drug development and regulatory science. We do all sorts of biomarker, regulatory biomarker development or drug development tool development. And these can be generated, we focus them around therapeutic areas. That was the most logical way to set this up. We have cancer, neuroscience, inflammation and immunity, and cardiometabolic respiratory. We're now going into the women's health space as well. So we have a number of therapeutic tiers there. And I say that because I know this might be one way that you guys think about organizing NAMS, is it might be by therapeutic area, it might be in other ways. As I said, all of these projects are pre-competitive. They're all funded by the private sector, and we do try and file actual qualification plans with the FDA whenever possible. Sometimes it takes us a couple of stages of the projects to get there. But again, once you get by the numbers, so we've got 60 plus active partners right now in the consortium, 40 active projects, $108 million raised, and we've had 2 biomarkers accepted for qualification for use as a surrogate endpoint by the FDA, 1 qualified composite safety marker, 14 therapeutics that have advanced based on tools that we've developed within the programs, and we've assisted in 9 FDA guidances and 9 clinical tools that are being used for drug development. So we've got a pretty good track record with this, and we do know that some of the things that NAMS may need to face are actual regulatory qualification packages and development that way. And so we think some of the infrastructure and some of the lessons that we've learned within the VC could be very relevant for what may go forward. So what is the role of FNIH when we do these? Well, as you can see, it can pretty much vary. We can do all 6 of what you see up there, or you can take that a la carte and we can kind of do whatever's needed of us. We're really just there to plug the gaps that need to be between the federal side and the private side. So we can do governance, establish structures to make sure everything's running. We can do policy management, provide safe harbor for different interactions and that sort of thing. Program management, this we do a lot of, putting stakeholders, convening, making sure everybody's on board. We do that quite well. Fundraising and relationship management, we can go out, bring the private sector that funds to the table, steward those funds. Project management, that's just the day in, day out. Everybody knows what that looks like. And we can't even do intellectual property management. We do try and keep in the pre-competitive space, but for each of our partnerships, they demand something slightly different. And we have at times held IP. We, for those who are familiar with the iSpy trial, iSpy 2 started with us. We ran it for the first 5 years before launching it out to Quantum Leap. We held the IP for that and the IND for that trial for the first 5 years. These aren't things that we do on a regular basis, but these are things we can do when necessary. So I just say all of this to kind of keep things thinking in the art of the possible. So a potential NAMS PPP. So Margaret set the stage beautifully for this. We have no We are not working cross purposes. We have no desire to, work contrary to what complementary is doing. We wish to kind of bring the community to the table for this. It is going to follow kind of this general trajectory and goals of what complementary is after. And really, this is to bring all kind of folks that are not within the federal government to the table to work with what complementary is doing. And as you saw from both my title and Margaret's notation, we're really going to be, we think, and talking with NIH, that our sweet spot will probably be the validation and qualification network portion of complementary, and where we think we can really recruit the private sector and have the most effect there. So I won't need to go over that. Sorry, normally Margaret doesn't usually get to talk right before me, so I usually have to have a brief interview before that. So what we intend to do, and what we've already put in process, is we're setting up a steering committee to act as a steering committee for the VQN. At least for the private sector side, we're inviting all the public sector members to participate in this conversation. We're going to work to collaborate with complementary. The only reason we've started ahead of this is, for those who aren't familiar with NIH process, there is a formal way to engage us when NIH wishes to. It's called a request for collaboration process. It has to go through a number of level reviews, including all the lawyers. So we wanted to get a jump start in the hopes that all of this gets approved and we move forward. So we're actually working. We had a kickoff meeting just to kind of solicit interest from the private sector, from manufacturers, from pharma, various places. And out of that, we are doing follow-up with two very simple questions. Are you interested in staying in the discussion? And two, well, I guess there's three. Two, are you interested in helping us support, because we are soliciting some private sector funds for the design phase? And then how should we set up these working groups to best move us towards our goal? Which I will admit, we've gotten about 10 responses and everybody's got a different opinion on how these should be set up. So if you've got opinions for me, please send them my way, because I think that'll be our biggest challenge, is figuring out how we break this down. So the point of the steering committee will be for putting together those working groups to evaluate projects, developing the proposals, and then taking those into full project development, and then assembling the funding and partnership for whatever it is that we choose to move forward. For the design phase itself, you heard Margaret talk about that year of design phase. We hope to be a part of that design phase once the RFC comes through. We're already holding conversations, as I noted, with NIH, FDA, potential partners. We would assemble all of those partners together to put together essentially working groups that would then discuss. We would put together what we would think to be use cases. All of that would culminate in kind of a white paper that we use to detail out to the rest of the world. This is what we think we want to do. Is this something that you're interested in joining us for? And we use that white paper as a form of solicitation to bring what we lovingly refer to as the coalition of the willing. And once we have the coalition of the willing together and we've all agreed on what the project should look like, at least for the first phase of whatever it is that we're doing, then we work to put together an official research plan that merges whatever, the federal side wants to do with the private side. And then once we have the funds raised, we can work alongside. NIH will control NIH's money. We will help to shepherd the private sector money, whether that is putting it in pots with NIH funding or us issuing grants ourselves, because we have that capability to do grants, contracts, grants, whatever is necessary for this. So again, for most of you in this space and who have been following the complementary, none of what's probably going to be on these next slides is very, very shocking to you. But we know that we need to develop some success criteria, establish minimum information for each of the NAMs that would want to go through qualification. We need to develop a governance structure. So what do we plan to do? The network, the FNIH, industry, NGOs. Identify primary kind of customers and associated regulatory agencies. So if we are focusing on the qualification portion of this, like, are we focusing solely in the U.S. to start out with, and then going to EMA, and then PDMA, and everybody else, or who else do we need to be? Is it other regulatory agencies? Because maybe we're not doing biomedical, maybe it's EPA, who knows? This is what we need to decide. Construct workshops and other activities to identify, industry and agency priority needs. We're going to seek feedback and scope on the validation and qualification efforts, define pre-competitive, and then we can basically issue RFPs to then solicit nominations for these late-stage NAMs to go in once we have actually identified what it is that we wish to focus on. And we do have the capability for competing RFPs. We can actually do it through a much more streamlined process generally than our federal partners, because we can limited compete and sole source and things like that if we need to, because we are in the nonprofit space. So this is just a much kind of more detailed, and I think these slides are going to be sent out, so I won't go through each of these, but this is just what we view in more detail. As you can see here, just a few things to highlight. Okay, a few things to highlight is that obviously the list of folks that you've seen in many other slides today, all of the federal partners, ICFAM, the 3Rs Consortium, IQMPS, HESI, CPAS, everybody. We're big tent kind of people. Eventually, some people might fall out of the tent. They don't want to stay and work with us. That's fine. We start big tent people, and then we'll work down from there. And like I said, what this design phase does is it's a white paper development, an aggregation of interest and scope, an advertisement for inclusion. It's a baseline for negotiation as we go out to the private sector partners. And then we get fairly tactical and granular in the white paper. We talk about cases, use cases, timelines, and budgets. And the budgets is the biggest thing. Like, what are we actually soliciting? We know what the federal government's going to put forward. What do we then need to ask of the private sector, whether that's in-kind dollars, that sort of thing, to know, to fill the gaps, to make sure we have a cohesive partnership. And then, you know, the milestones and kind of expected outputs, that's where once we've had the coalition of the willing, once we know what the resources are on the table, we can get that granular and kind of put that together for a very structured research plan. For the design phase itself, because we are going to be working with complementary, or at least that's the hope, they are working with us, and they have been kind enough to agree to kind of half the design phase with us. And so they're working across the agencies that Margaret showed to put forward about $300K, and we're working to get the other $200K or any additional funds that we may end up needing. So any additional things beyond the 300 will come from the private sector. We've just estimated that it'll be about 200. And so we are in process of soliciting those resources now from the private sector to get started. And what that covers is, you know, time. So we get a dedicated project manager at the SNIH that will oversee this, any in-person meetings that we need to host, travel that we need to do. Do we need to go out and meet with folks at meetings or things like that to make sure this is moving smoothly? So that's what this costs. There aren't any kind of tangible products that come out of this other than the white paper. This is really just you're getting a dedicated resource to make sure that this moves forward. And then the implementation phase plan, those are all ballparks, and this kind of comes from our conversations around complementary, but we don't really know. I'm just going to say that. Those are because we don't know. It's going to come in the design phase. That's why we run the design phase. You run a design phase to know what your budget is for the implementation phase. So people kept asking us, well, do you have a ballpark of what we're looking at? And that was the best thing that we could kind of put up with once we had gone through. As I said, this is where we're at right now. We're trying to decide how to assemble the working groups to best complement what complementary is wishing to move forward with. As you saw, you know, obviously they're talking around technology and chemical and silico in vitro. We could go by sector. We know biomedical and chemical and environmental look at this very differently. Maybe it's around use case, maybe it's safety, maybe it's toxicology. Maybe it's around therapeutic area. As I said, our biomarker consortium, where we do a lot of regulatory work, we do it around therapeutic verticals. So we're not really sure. So we're actually looking for some really strong recommendations in this capacity. Like I said, we've solicited from the folks that we had at our kickoff meeting. I've had 10 responses, and all 10 responses have been different. So I'm trying to look for some consensus, and maybe it's a bit of a matrix. Maybe it's got to go by technology and therapeutic area. I will say that probably any more than 6 to 8 working groups gets a bit unruly. So that's what I'm trying to keep it to. So we are going to matrix these things, want to just keep it manageable. So next steps, we're polling for feedback, soliciting support from the private sector. Throughout Q4, we're going to work to establish those working groups. Everybody will get to self-select membership as to what working group you want to be a part of once we get them up and running. We're going to re-engage the full steering committee, which we had a kickoff for in July, every couple of months. And then as soon as we are fully papered and signed, we're going to, you know, work very closely with Margaret and her team on complementary just to make sure that, you know, we're working together and working to the best interest of both sides of the equation. And then we'll begin the design phase in earnest fingers crossed, in Q1 of 2025. So that's all I have, except for to take any questions for clarification.
Yeah, clarification questions.
There, Mike. There you go.
Hi, Chantel Nicholas from App Global. I was wondering about this coalition of the willing, or COW. I was wondering, does it really focus on including entities that are sort of paying in, or does it also have entities that just have great ideas?
So it can kind of go either way. It really depends on the size of the organization and what the organization's purpose there is. And we also, in kind, don't always think hard cash dollars, although hard cash dollars are nice because they're the most flexible to do whatever we might need to do with. But a lot of times when we're talking about participation in the implementation phase of a project, that coalition of the willing, it will be tiered by organization size. For the biomarkers consortium, I'll use that as an example. We do it by R&D budget. And we have a baseline that, you know, if there's a nonprofit or a professional agency that wants to participate with us, that's like a even lower tier to come in. And oftentimes it's, you know, for membership at the consortium itself, that's like $5,000 a year. It's, you know, we try and keep it at a price that we think is affordable, have everybody there at the table. And then there's also, we have the discretion, and this really comes with the discretion of the members of the coalition or the willing or the COW, to bring in subject matter experts that is their entire job. They need to be there because they have a voice, they have that sort of thing. We've had policy implementers sit at the table. You know, oftentimes our regulators don't contribute money. What they contribute is expertise, and they do it as a non-voting member because that keeps them out of conflict of interest if they're going to review anything that's coming through. But so we're very flexible. We just try and figure out how the partnership has to come together. I'd love to say everybody can participate without paying, but we just know that's not practical. So we just try and figure out where we can get the most engagement across the board, and it's going to make everybody happy. When I took this role 8.5 years ago, my boss at the time said, look, you'll know you have done your job if you get everybody around the table and you all have the conversation and you agree on something. Everybody gets slightly disgruntled, but they're always willing to come back and talk to you again. So that's the goal.
So thanks so much, Stacey, for the excellent presentation and overview. And I wanted to build off of, you said you kind of spoke just a little bit in your last answer, but if you could expand at all on some of the lessons that you all have learned over the last decade from the AMP and the Biomarkers Consortium that sort of could be applied to the design phase of the VQN. I don't know if there's anything that really jumps out at you that you could share with us. That's the first part of my question. And then the second part of the question is, I think data sharing will be absolutely germane to the success of this initiative in the VQN. And so I wonder if you have any thoughts about sort of the appetite for data sharing or the willingness to share data in the private sector, and whether or not there are creative approaches to using things like encryption technologies or federated model building that would allow us to better facilitate willingness towards data sharing.
Yeah. So how about I start with the second, and we can go back to the first. So because we can get broader. So for data sharing, we've got a lot of different constructs for data sharing as well. Many of the AMP portals are patient level de-identified data. I'd say probably seven of the 12 are, because 2 of them haven't yet constructed their portals. So 7 of 10 are patient level de-identified data. And I'd say about half of those, the data all comes from maybe NIH programs, observational cohorts, that sort of thing. So it's not so much of an issue, even though there might be proprietary data in there because they're observational cohorts, there's no intervention and there's no really touched subject. However, the other half actually have interventional data in them. And some of that comes from NIH-based experiments, some of it actually comes from companies. and they've been willing because in certain areas, especially in the Alzheimer's space early on, think about a decade ago where we were with Alzheimer's research, companies were running the trials, but nobody was having any success. And so everybody kind of needed to pool the data to figure out, like, what do we need to do? How do we mine this? Why aren't certain things working? And so I would say that there can be an appetite. It has to be the right area. And so this goes to, I guess this will go to my first general lesson is, when we look at this and when we're looking at picking projects to do as a public-private partnership that isn't just going to be a grant award that maybe comes out of complementary or that comes out of a single agency, it has to be something that can't be done under a single one-to-one relationship. It has to be something that nobody else is tackling, or at least nobody believes that it's being tackled well. We do very well when we can find our niche. And it's also something where, can we select something? Can we select a project such as a standards development project or something like that, where no one person can do it alone, nothing terribly, you're not generating anything proprietary out of it, but it's going to move the whole field forward. And I'll give it a biomarker example. We do a lot in the hematology, oncology space, and we've been very successful in MRD. We had a first project in ALL. We moved into multiple myeloma. We then moved into AML. And we've had the AML project now, we have 10 diagnostic companies working with us. We're not validating their technology. What we're validating is the biomarker of MRD. That's what FDA is going to qualify as a certain level of, measurable, detectable disease. And we don't care what technology it is. And so if the companies will come to us and they'll work with us and we can figure out what technologies work and that will all work at the same level of a 10 to the negative 6 cutoff, it behooves them because their technology then can move forward. They can use the data to move forward with FDA. What we care about is that all the data goes. So if you can find something like that where you can kind of get consensus, I don't want to call it generalizable, but, value add across the board. As far as going back to the data sharing question, even beyond us, even, thinking outside the foundation, I will say there is another project in the cancer space called Project DataSphere, in which private sector companies have actually donated the placebo arms of all of their trials into a single database to be mined against each other for kind of standard of care use and that sort of thing. And Martin Murphy and the Project DataSphere, I think Sean Cozen, who used to be head of the real world evidence at FDA, he now oversees Project DataSphere. And so I think there are models by which this can be done. I think it just kind of have to hit the sweet spot of, once again, a problem that you need everybody's data to solve and that no one person, and it's going to benefit everybody to solve it. And so the other side of the AMP equation, the ones that aren't patient level identifiable data that sit in the portal, 2 of them we actually run as a federated model. So type 2 diabetes started it, common metabolic disease continued it. They're trial level data, but we pull them from across the globe. And we've even had to deal in the realm of GDPR. And for those who don't know, the federal government can't actually sign on to GDPR. It's a whole thing. So we had to set up a whole federated node in Europe. We work with the European Bioinformatics Society and have a federated node there because some of the best data sets in type 2 diabetes happen to come from the Scandinavian countries. And so, so once again, we can get creative as long as we think we can get all the right partners to the table and there is a desire to do it. I think that there is an appetite that we can get it done. That's helpful.
My question is easy, but that was amazing. I was so mesmerized by your talking, and I was like, what was I going to ask? You, in your big matrix, and I know I've seen that because we've been looking at different ways that you could look at that. When you have the usability context, you have safety and toxicology, but there's a lot more there, right? Like we potentially can add in. I was just thinking
100%. Like we were just trying to give people ideas. I will say we never have a horse in the race. It's the consensus of what everybody wants. That's what we move forward with. We have enough knowledge to be dangerous in most spaces that we play in. My background is in cancer. It's definitely not an alternative animal model. I actually worked on animal models. So again, this is a whole new space for me, but we really just want to listen to everyone. So if you have other ideas, if you think there's other things that belong there and that's the way we think should be setting up, go ahead and email me.
I'll send you some ideas. I think just one, and since, I know these are clarifying questions, but one of the things I've found interesting is people that work in the safety toxicity space, it's very scary for them to go into this new technology because if something goes wrong, they feel like it could be a major setback. And so then, do we just look at mechanism of action, some other kind of things that in that. So I'll send you suggestions. So thank you.
I am not seeing any more clarifying questions. So we are, for real this time, going to move on to the next session.
Everybody can find it.
Thank you so much. Okay, so we're going to move on to public comment section. So all public comments were solicited through the Federal Register notice. Written comments were posted to the meeting webpage and distributed to all SACATA members in advance of the meeting. And individuals requesting to make oral comment were required to register in advance for planning purposes. So I'm just going to acknowledge that there were 3 written comments for this session that were received. One was from the Humane Society of the United States. One was from Physicians Committee for Responsible Medicine. And the other one was from People for the Ethical Treatment of Animals. And there were 3 oral presentations, oral presenters registered. And I would like to ask, the first one is Christy Sullivan, and she is remote. So I'll have her start.
Thank you, Dr. Page. This is Christy Sullivan with the Institute for In Vitro Sciences, as you said. So we had a spanning lunch. We had some really nice and stimulating presentations, and I will offer some comments on a few of them.
Christy, your audio is breaking up a little.
Okay. Actually, let me just turn off my headphones because that's the new thing from this morning. All right. Is that better?
Yes.
Okay. All right. So first of all, I just wanted to briefly address the Method Developers Forum. I think this is a really exciting initiative and process that you've put together to try to make the forums very productive and interactive. And I think it will help to connect regulators and developers to help facilitate implementation of new approaches that are kind of out there being developed. I support some of the lessons learned that you presented, Dr. Rinke. I think those are good ideas for moving forward. A couple of recommendations I also have come up with, which is I would see about trying to make more time for discussion and maybe consider something like this SACATA meeting where you have charged someone from the agencies to sort of either ask questions or make comments or have a response so that you have some more interaction. I would also think about providing a slide template with characterization criteria that might help structure the presentations. And I felt like some of the presentations were very sort of scientific conferencey and kind of get them moving towards something more focused on agency needs. It would also be really nice to hear feedback from the participating agencies on any outcomes they've undertaken as a result of any of the method developer forums. And then taking a page from the OECD IATA case studies program, might explore sending agencies a post-forum survey that asks them to talk about how they might use the methods that were presented, or if they wouldn't, why wouldn't they? Is it a matter of the limitations of the method? Is it lack of applicability for their own needs? Is there some kind of science barrier, policy barrier? I think the OECD started doing that a couple of years back for some of the case studies that were presented, and it really helped to jumpstart some discussions and help the method developers think about how to make progress. The rest of my comments, I'm going to talk about validation activities and complementary projects all kind of together. First of all, I want to say that I happen to work with NICEDM, ICFAM, and innovative scientists on 3 OECD projects that are ongoing. And I know that they're working on several others. I think the innovative and NICEDM team in particular is really helping to drive progress at OECD in order to achieve that international harmonization of some of the ICFAM agency priorities. So I just wanted to give them a little bit of a kudos. Charles asked the question, how can we leverage ICFAM to inform OECD? And one thing that came to my mind was for ICFAM agency scientists to consider getting more involved in working party on hazard assessment activities. This is a different working party than the WNT. They have some other activities going on. They have the OECD IATA Case Studies project. They have create guidance for applying and evaluating new tools in a regulatory context like PBK models and QSAR predictions. And again, that IATA Case Studies program is particularly useful as it creates a forum to help test and discuss NAM-based strategies for regulatory application. I also want to say thanks to Drs. Gadbois, Otrochinska, and Adam for sharing the really exciting program updates. I'm really, it's so exciting to see so much investment and thought going into how to validate and develop and validate new approaches for biomedical science. For both toxicology and biomedical science, I cannot emphasize enough the need for funding for validation and regulatory readiness activities. I mean, that's already been mentioned, but it's really, really important. Regarding specifically the complementary program, wanted to encourage a balance in the funding dedicated to the technology development centers on the one hand versus the validation and qualification activities. In our experience, validation is a really iterative process where you have development and then evaluation, optimization, back to development. And so either to make sure that process goes well, we encourage more funding be planned for the validation and qualification activities, or to ensure that validation expertise is housed within the technology development centers themselves. Funding, of course, includes, but is broader than the validation that we're all thinking of, the sort of parallel testing of accuracy and reproducibility. There are other activities like tissue shipping studies. Do the tissues work when you ship them around the world? Equipment calibration, testing of the protocol, kind of ensuring reproducible processes in the laboratory. And I encourage some of these activities to be included within validation planning. Funding is also needed for capacity building as we envision this transition towards more and more in vitro and computational approaches. So small and large labs will have a hard time bringing on multiple new assays at once to be able to offer, for example, just to take one example, the DNT, developmental neurotox battery, because it requires several new pieces of equipment. Unless we have some confidence that the assays will be requested and accepted by regulatory agencies, it's difficult to make investments like that. We could think about this issue being addressed with a loan program or a partnership or a center program, maybe in line with some of the presentations we've seen already. And then finally, another urgent need besides funding for validation success is expertise and guidance. Those who have experience in method validation can share learnings to ensure The validation activities are efficient and well-planned. Hearing Dr. Adams' presentation, we encourage outreach to organizations that have been engaged in NAMS validation for regulatory application for the past couple of decades to ensure that all of our collective learnings as a field are built upon. And I could also even envision a coaching process for individual developers built around the good in vitro methods, practices, principles. Those were my comments and suggestions. Thanks very much for the opportunity.
Thank you, Christy. I'd like to open it up to any clarifying questions for the presenter. And I'm not seeing any, so we are going to move on to the next oral presentation from Megan LaFollette from the 3 Hours Collaborative. Okay, so I think that we may be having some trouble contacting Megan for a second. Maybe she just stepped away for a second. So we're going to, okay. So while we sort that out, we're going to have Shagun Krishna from the Physicians Committee for Responsible Medicine come up to the podium, please, and present your oral comments.
Hi, good afternoon. Can you hear me? Okay. So thank you for the opportunity to speak today on behalf of Physicians Committee for Responsible Medicine. My name is Shagun Krishna, and I would like to keep my comments brief as we have already submitted a detailed written comment. And first, I want to commend the updates to OECD Guidance Document 34 provided by Dr. Kovac. It's great to see how these revisions are adapting to accommodate the NAMS and defining approaches for the technical validation as well. These updates are essential to pushing the non-animal methods forward, particularly in regulatory testing. Clarifying terms, readiness criteria, and defining the technical validation process in the presentation is really going to help streamline the NAM adoption. And we also want to express our support for the Method Developers Forum. The forum is doing such an important work by bringing NAM developers and stakeholders and regulators together to work through the challenges and accelerate the validation process. And we feel that it's an excellent platform that's going to help moving the field forward. And personally, I'm really looking forward for the next forum, which is going to related to the cardiovascular endpoints. And we also want to express our, we are really excited to see the efforts in catalyzing the development and use of NAMs, especially through the recommendations from the NIH Advisory Committee to the Director Working Group. The vision for developing patient-specific NAMs and integrated approaches is highly welcomed, as mentioned in Dr. Gedboy's presentation just like an hour before. And we do believe that the complementary program is an incredibly exciting initiative. It's focused on developing human-based models to replace animal research, and that's a critical step in making research more reliable and ethical. And I'm really looking forward to see that how this program is going to shape the future of biomedical research. And we also appreciate the public-private partnership led by the Foundation of NIH. They play a crucial role in accelerating the validation and broader adoption of NAMs. And it was really great to see the progress of design phase activity of VQN, as mentioned in Dr. Adams' presentation. And we also want to encourage Psychatom to keep supporting efforts to harmonize regulation across different agencies. Regulatory harmonization is very important for making sure that NAMs are adopted consistently. Clear communication to industry stakeholders and formal policy updates are going to be key to building confidence in these approaches. We hear a lot about companies who really want and are interested in adopting NAM, but they are just hesitant because they are very uncertain about the regulatory acceptance. And finally, as discussed earlier today, we want to stress the importance of leveraging human relevance data in NAM evaluation. It's pretty obvious that human data gives us much more accurate insights. And I think CICATM and ICRAM agencies can really help by facilitating partnerships to share human data for validation purpose. And one way to achieve this could be through partnering with All of Us program, as discussed today as mentioned by Dr. Wojcik earlier. And before I wrap up, I just want to take a moment to thank CICATM leadership for their continued commitment in promoting non-animal methods. And thank you again for providing me the opportunity to share my thoughts. That's all.
Do we have any clarifying questions? If not, we can move on to Megan. Are you able to unmute yourself?
Yes, I think so. Can you hear me now?
Perfect. Yes, we can. Excellent.
Okay.
So we'll project the slides.
Sweet. Thank you. Thank you for your patience. Always wouldn't be a true hybrid meeting if we didn't have at least one technical difficulty. So cool. Okay. Can you guys see my slides?
Are you sharing from your screen, or do you want us to share from here?
I don't think I have the capacity to, I don't have the share button.
Okay, I'll pull them up and share them from here.
Yeah, great.
You should be able to see them now.
Awesome. Thank you so much. And yeah, again, thank you for bearing with me. My name is Dr. Megan LaFollette, and I'm Executive Director at the 3Rs Collaborative. I want to note that our current president, Dr. Sally Thompson-Iritani, is in the room in person today, and I believe she'll be there tomorrow. So if you do have more extensive follow-up questions about us or the public comments, feel free to talk to her. Next slide. So next slide. Awesome. So just real quick, a little bit about us and our organization. So we're a US-based nonprofit whose mission is to advance better science for both people and animals. And we achieve this goal through facilitating collaborative 3Rs efforts focused on refinement, reduction, and replacement. Next slide. And on the next slide, you'll see that we do have some shared interests and goals with the potential for future partnerships. And you can go ahead one more slide as well. Sorry, I thought I might be able to control today, and I've got kind of a lot of slides. Awesome. So yeah, one more slide. So we have a lot of different efforts that span across critical topics. They go both from refinement to reduction and replacement. Of most interest today would probably be our initiatives that are in translational digital biomarkers, microphysiological systems, and artificial intelligence. Although I'll also note that not pictured on the slides, we also have an initiative focused on creating a 3R certificate course, as well as compassion fatigue resiliency. Next slide. To go on to our MPS initiative, it is quite a large initiative. It's comprised primarily of commercial developers. It actually includes 42 institutions and 30 developers. Next slide. And we do run a tech hub if you want to learn more about these commercial providers that features all of them and actually allows you to filter them by their tissues, disease models, and species. So, you know, like FNIH was talking about how to divide up different companies, there's many different ways. This is one way. I encourage you to check that out. Next slide. And after going through that just really brief about us, I do want to talk about three kind of commendations and recommendations. And this is really for CICATM as a whole, not necessarily just this section. So the first is we both commend the current efforts and encourage you to continue to enhance collaborations between both government and non-governmental agencies. We especially encourage this with organizations that are balanced across all 3 Rs and encourage collaborations between researchers that are currently using animal models and or NAMs in various sectors. We think that this balanced approach and actually working together is going to make the most implementation in the fastest amount of time. And in particular, we do really want to commend the Method Developer Forum. This is something that certainly I will be keeping an eye on and making sure that I'm sharing information about to our commercial MPS developers and throughout our network. And also, we really commend the work that the Advisory Committee to Director Working Group in catalyzing the development and use of novel alternative methods. Their report is a really great resource for the field. I think it's really comprehensive and just really does a great job outlining everything for the field. Next slide. One additional, and then I think there's one more animation that you can click through. A recommendation for Cicada members would be to learn more about some commercial MPS offerings. We have a formal MOU in place with the IQ MPS affiliate, and we've run a number of these data-driven workshops that has 15-minute presentations from developers. So of course, love the Method Developer Forum that you guys have, but potentially in preparation, if you're going to do organ specific, or now we've been moving on to like disease area specific, it might be useful to review these in advance. Maybe there's a potential for a greater partnership on this. And then I just want to mention that we have 2 upcoming ones on cell sourcing and oncology. One is actually next week. So love for any interested parties to join those, and you can find that on the website. Next slide. And I just want to showcase that these are all recorded. They're on our website and YouTube's for a really easy reference for you to find them. Next slide. I also want to commend Cicada members on the realm of collaboration for participating in our artificial intelligence initiative that is focused on preclinical safety and risk assessment. We've got a number of different members on this. We are really, really thankful for their current participation and hope that continues and encourage more in the future. That's a pretty early initiative. We're working on a kind of comprehensive review paper, but are excited to kind of see what goes next. Next slide. Second kind of key point is just to both, again, commend and encourage continued investments for independent characterization and validation of NAMs, especially those that are commercialized, because we do see that those are going to have the widest access and are often the most fully along in their development. We're really excited. I've been one of that 10 responses that gave a different response to Stacy at the FNIH, but we're excited to participate on that NAMs validation and qualification network steering committee. And we also have a partnership with the FDA CDER, and again, commend that. Next slide. And I just want to highlight, again, this is both like a commendation. The specific project is early, but we are doing this with the FDA CDER. That is a cross-platform project on MPS for drug-induced liver injury. It's got a lot of the same goals, I think, as the FNIH BQN, NAMS BQN. And so, potential partnership on this, I think, could be great in the future. I think the more that we all learn about these different projects, this is really cross-platform working with the different developers. We have a rep from FDA CDER. So we're excited to go forward with this. We'd love to work with FNIH more on that. So yeah, just wanted to highlight that. Next slide. And then just like the third and final, both again, commendation as well as recommendation, is just to continue to endorse and invest in furthering the implementation of these scientifically supported NAMs. Again, we really encourage using kind of balanced, accurate messaging to promote positive attitudes, provide training and education, and assist in normalization and socialization of the 3 Rs. This is something that we really focus on our organization, that people who currently use animal models now are the people that we often want to use NAMs in the future. And so we want to make sure that our messaging is really speaks to them and doesn't immediately turn them off so they stop listening. And again, the complementary program, the common fund work, as well as ICBAM's development of different programs like Opera and ICE. There's been a lot of great work done here, and I'm really excited to see how it continues. And then, yeah, the last slide that I've got is just if you are interested in following along with our organization, we've got a newsletter. Like I said, we've got a bunch of resources on our website, and I'd always be happy if you email me, talk to Sally in person, and I'll take any questions now. Thank you so much for the opportunity to comment.
Great, and we're going to open it up for any clarifying questions. Dr. Sura, go ahead.
Thank you, Megan. A quick clarifying question. Can you elaborate a bit on AI and its use in preclinical safety as well as risk assessment? You mentioned about the environmental cicada. I just wanted to understand a bit more on that, what you meant and
yes.
How AI is used. Thank you.
Yeah, absolutely. So this is still a pretty early initiative here at the 3Rs Collaborative, but essentially what we've done is we've gathered stakeholders from a couple different agencies, industry, tech providers, and what we're trying to do right now is just write this review paper about the current use of AI in preclinical safety and risk assessment that's fairly broad, that's fairly high level, and addresses both why is this so important right now, as well as what are some key challenges going forward. That review paper is coming first, and then from there, we'll kind of move to more targeted goals. And so I know that there's various members and maybe I misspoke, maybe it's different members of ICVAM versus the CICADM committee that are, there's some overlap there. But yeah, I know there's some members that are already involved in that initiative, and once we get this initial group paper out, we're going to be opening that group up for further participation from more members, and we'd always love just more subject matter experts to join that initiative to help us lead on that topic. I don't know if that answered your question or if you have more specific follow-up questions as well. Happy to take
No, it's good. Thank you.
Okay, thanks.
Okay, I am not seeing any additional clarifying questions at this time, so we will move on to the scatter discussion portion. So remember that we do have two parts of session 2, A and B. So we're going to start with the validation updates, which was section A, and we'll have the lead discussants, starting with Dr. Barron. Go first, please.
Great, thank you so much. I really appreciate having an opportunity to be here. I'm also sad that I'm not there, and also sad this is my last year with the committee. But I also want to add, and send kudos for all of the work that happens within a year. I mean, the deliverables, they're already presented today, and there's going to be more presented tomorrow. It's pretty incredible how it drives that to those technologies forward. So again, just want to, in the report, also, if you didn't have a chance to read that, I would highly suggest to read it in quite detail, because that's something that really outlines all of the different projects that have been completed here, which again, kudos on those. So first I want to address the first question here about the OECD guidance. And then so one of the things that I think when those are developed, and very often when we talk about NAMs, a lot of it seems to be geared towards or easier to apply to in vitro NAMs versus, for example, like in silico NAMs. So really, I think making sure that through that guidance to consider both of those technologies and others as well. I think also when we think about the in silico aspects, looking at simulation versus prediction technologies in that space, because that validation for those are going to be quite different. So I definitely don't want the guidance to be prescriptive, but being more clear on what they would look like, I think really would benefit those technologies moving forward. What was already mentioned that the validation is very often based on an existing models and methods which exist. And in a lot of cases, especially when you're looking at novel, for example, endpoints, the models don't exist, right? So again, it was already mentioned, but just wanted to second that. That's imperative that that's addressed within the guidance. Also, considering validating output versus the technology, right? And that was already mentioned with the biomarker consortium. That's how they approach that. Obviously, that comes with its challenges, but I think that as we're moving to some of these methods, that would expedite the validation process. Also, what was mentioned is that publications play a significant role through the validation aspect, but it also presents a challenge because very often journals or reviewers ask for data or information that technology providers cannot share. So for example, like the code that comes with it. So how to address that or maybe discuss that with the journals and the reviewers moving forward. And also the transferring the AI tech between different labs, that's a challenge. Again, that was brought up earlier, but that's really a significant challenge when it, for example, comes to IP and others aspects as well as the technology is being implemented. And again, I mentioned that, don't expect this to be prescriptive, but it's adding additional clarity on the validation aspect is going to be imperative. Also, sharing the use cases of the validations technologies and all the details that come with it. That would be very imperative as well. And also considering validation of combinatorial names. So when you combine, for example, NPS with AI technology, validating each one of those separately is a challenge enough, but how would you validate if they're combined together? And then kind of moving to question 2, 3, and 4, kind of pull those together. One, funding for the validation. The validation aspect, this seems to be always really, I guess, undercounted on what it takes to do that. And increasing the dialogue opportunities for the technology providers and the end users. The other aspect, which is already touched upon, is the data access. I think that's one of the biggest challenges with these technologies. So how do you address that? And there was already some aspects put in here and solutions suggested, but doing that in a collaborative manner. So also, what would be really helpful is developing a framework for return on that investment, right? And including specific metrics or developing a calculator for these technologies, because that's where the engagement, very often technologies are validated and even being accepted by regulators. But as we know, some of them are not being implemented on routine basis. And one of the challenges with that is that identifying what's the return on an investment with these technologies and using those technologies. Including non-governmental stakeholders in the publications from CICATM, not just to kind of taking a step further beyond just the surveys and then putting that information in the manuscript, but bringing those other stakeholders into the publication. Again, I know that becomes also challenging, bring herding all the cats together, but I think that would be imperative moving forward and would really increase the engagement with these technologies. Education and training, that was already mentioned. That's one of the recommendations that came from the NIH report on alternatives. And that's something that CICATM is already doing, but just stressing that that's really important and spreading the information, all the technologies that are available and what has been validated, what hasn't, would really help. To continue updates, making them available. There's discussion about the website, which again, just kudos to the website, the way now you can go and search from the different organizations that are participating in there. And I know it makes it really challenging to make it live. That was already brought up before. And also initiate and increase the cross-industries collaboration on these technologies. So the chemical industry versus, and pharmaceutical industries as an example. And across all of the FDA centers, because many of them are also working on these technologies, but in a different spaces, obviously. So increasing that collaboration and transferring the learnings from each one of those groups, I think would be helpful. Validation versus implementation tracking. So different things are going to be validated, but again, kind of what I mentioned earlier, are they really truly being implemented? And can we track that and share that information? Also, one of the things that the benchmarking, and this kind of goes to the validation, I'd be curious, how Cicadium is going to address this question. But so are the in vivo model, to a certain extent, is a black box, but it's accepted, right? Because we've been utilizing for so long. But then in silico models, for example, or even some of the NPS models are also a black box to us, right? But they're not being accepted, even if there is significant amount of data providing it. So I think from a validation perspective, we'll be interested to see how that evolves. Also, for the fit for purpose framework validation publication, it includes one of the aspects that talks about using the technology for the current endpoints, regulatory endpoints that are there. But with these technologies, obviously, we're going to be looking at novel endpoints and biomarkers. So expanding that and including that there. I already talked about, just from my notes here, just increasing the live dialogue between tech providers and end users. I think one of the presentations, this afternoon, they were given from the NIH. So really increasing the collaboration with NIH while implementing the recommendations from NIH advisory group of NAMs. And I believe, that's already happening to a certain extent, but really formalize their interaction. I think one of the other aspects for collaboration that could benefit is to collaborate on NAMs with the biomarkers consortium. So the way the approach they take to validate biomarkers, and there's a lot of earnings from the last 2 decades that I think we could take advantage of. And also, which I think, Nicole, you already mentioned, right, collaborating with FNIH during the design phase of the VQN, and specifically on the data sharing and data engagement. I think that's, again, like I mentioned earlier, right, the data, I think it's one of the biggest challenges for us and the access to the data. And overall, just, collaborating to sharing learnings among organizations and industries, which Cicadium is doing an amazing job at that, as it was shown today, and I'm sure it's going to be shown tomorrow. But there's not enough way. The collaboration is what's going to drive these things forward from all of the different stakeholders. So again, just wanted to kind of second that aspect. And so I'll stop there. Thank you again for the opportunity to comment.
Okay, does anyone want to respond? Just please, reminder that if we're using the Zoom feature, you can raise your hand so that I know that if anyone wants to respond, you can do it in the room, but it is very helpful for me. I'm not seeing anything, Stefan, so we will move on to Sue.
Okay, you hear me? Good. Sue Leary, member of Cicadium Alternatives Research and Development Foundation, and I'm also rotating off of Cicadium this year. It's been a pleasure to serve. I thought back to when I was invited to join, and Francis Collins was telling us how to wash our hands. So it was a while ago, and I watched it. He also played the guitar, doesn't he? Remember? Anyway, it's been a long time, and I will come to the next Cicadium meeting, so you can't get rid of me that easily. And I was just looking over some of my old folders, and I actually have minutes from a Cicadium meeting in 2003. And we were talking about the BCOP, the bovine corneal opacity and permeability test, and spending a great deal of time on that. And it was what I used to call the eye of the needle process of validation. Maybe gatekeepers is a little too strong a word, but there certainly has been tremendous growth and interest, and it's a very exciting time. I know that the questions don't really ask this, but I do want to commend the folks in the validation working group. That was an amazing report. I guess we talked about it a little bit at the last Academy meeting, but now the final version is out. It feels like it's really a new era. There's been a little bit of democratization of the validation process. I will say that I think the leadership of the working group is still needed. I think there's the key points in the validation report may not be entirely clear to all the end users and even the method developers. So I think that it would probably be very helpful for that working group to transpose into an expert group and make themselves available to individual agencies when they're doing their planning. I think that the more we have more ways in with some funding opportunities around validation would be very helpful and help advance things quickly. But it is a new era, and that started probably around 2013. And lots of folks around this table to be commended for their work, Dr. Kleinster and Dr. Casey. And I do think, though, that it's still important to monitor how some agencies are going to approach validation. I even recently heard of an agency that was going to run animal studies against the in vitro methods. Seems like they didn't quite get the memo, and it is intuitive to say, well, we have this animal test, we have a new in vitro method, let's run it side by side. But I think the whole new approach of evaluating test methods on their biological and mechanistic relevance is the way forward, and I think it's going to take a little monitoring and maybe a little time to be clear that that's the way to approach it. And I think in the end that will help us be more protective of public health. So just in general, I think what we heard was reported in one of the surveys. The two basic questions haven't changed too much over the years. What alternative methods are available and validated, and which ones will the agencies accept? That's basically the value proposition that everybody wants to know. So if we just, there's a lot of activities, a lot of very specific case studies that are building the body of knowledge, but at the end of the day, those are the two real clear questions that people want to know, and the more we can provide that information, the better. And for the agencies to become more involved, I heard several suggestions about that around the Method Developers Forum to help the agencies get more engaged in a dialogue. Because if the agencies are not using or encouraging the use of NAMs when they are available, they are not really practicing the 3 Rs. So I think that there needs to be more encouragement of their use, and the best way to do that is to make clear which ones they will accept, which has been mentioned for a long time. Our own little foundation, I will just share some information that we had more than twice as many applications this year for our little grants than we've ever had. So and we've been doing this since 1993. So I will say there's a lot of excitement out there among bench scientists who are really seeing the possibilities of using these. We have to scale up quickly and make some funding available, get some training out there. I like the translational centers for MPS. I think they're going to be very helpful. And it would be great to have some educational opportunities that are not necessarily associated with commercial entities, which are doing a great job, actually, of, they'll step in if the other education isn't there, showing how to. I read one protocol the other day, and I thought, boy, I think I could do this at my kitchen table. They make it look so simple. So I think the education is really important. To these questions, I thought the OECD approach, the slide with the priorities, the thing that jumped out to me was finding a way to validate the defined approaches. I think there's going to be much more that unifying the in silico and in vitro is going to be much more prominent, and that seems like a really good top priority. The method developers forum, as others have said, it just seems like a great way to move forward and help the validation effort. And the case study, I just want to commend the folks at DISC, and I loved the DISC Rev. That was fun, and I think there's going to be more and more case studies. That's what people seem to want to focus on. It feels right. So I think that's basically it for me. Good job, everybody.
Great. I am not taking any clarifying questions. Now we will move on. Dr. Benham.
Can you hear me? Great. Okay. Thanks. I think I can say I'm new here to SACADEM. I'm a faculty at the University of Pittsburgh. I've been overwhelmed, but in a good way. It's been very informative session, and I really found a lot of educational content here, especially if you're new to this area, and I think it might apply to a good number of people online. It's amazing that how the whole idea of SACADEM, NICEDM, and ICBAM has come together, and the agencies come together, and this great interest by all the stakeholders coming together. But it's a massive effort, and I think it has to evolve, and it's going to evolve more. So I'm just going to go through kind of some of my thoughts about the questions raised here. About the OECD, I think the sort of things that would help was expediting the process, which was kind of the core of the presentation, which is very good. But another aspect is that if the whole idea is to create guidance, to have platforms that are going to be used for toxicology, especially chemicals, we need to bring biologists engage. And my feeling is that maybe we can do a better job to engage with the biologists, that they don't necessarily have a stake in chemical toxicity, but their science can push the field forward. In other words, there might be a lot of groups, generally I'm talking about academic labs, that they do a lot of development, innovation, but they can support these efforts, but they're not engaged directly with toxicology. So maybe we need efforts to kind of engage those folks more to accelerate NAMs that can be actually applied for that. Expediting the process, which was mentioned, I think that would be very helpful. And with the model of the NAMs, whether we talk about in silico approaches, whether we talk about MPS, I think we need to have a clear value proposition. Just because a new model comes out, it doesn't mean it should count as a NAM. We need to understand what value it offers that is either better or solve the problem from existing approaches. And for that, we need to be very brutally honest to ourselves. As we innovate, we develop this, And we need to have consensus in the field, whether in terms of application toxicology or it's basic science or anything, to say, okay, this is the real value of this model. And then we try to apply it for the context of application to see how they can be pushed forward. It would be beneficial, especially in the context of OECD and methods developer platforms, that we boost innovation. Sometimes we move towards to have a single universal, for example, platform that solves a lot of problems. But we need to understand not all the time one size fits all. So you need to have continued innovation to kind of support these efforts. In other words, coming from the problem and finding solution might be a better approach rather than saying, let's have a universal approach that predicts all the inhalation toxicology problems. Let's find out what exact inhalation toxicology problem we want to tackle. Where is it that animals fail and how we can tackle it? Then the model can be developed, even if it's not existent at the moment. I really liked the presentation by Amy on inhalation toxicology. That was wonderful. And I think it's a great approach that she tried to narrow down to sort of see the papers have been published, what is it has been done, etc. Again, when you want to bring in the biologists and scientists that they have no stake in inhalation toxicology, but they care about the model, I think it comes that when we draw the barrier, when enough is enough in terms of biological complexity. We cannot have ultimate complexity. And we don't need to have ultimate complexity for every organ. In that case, we just need to have an actual human to test it, but that's not possible. So I think we need to be able to basically have consensus and create a framework that when we cut it in terms of biological relevance, biological complexity, and create a stepwise approach to kind of build these platforms. And the same idea could potentially apply to other fields of toxicology as well. What would be most beneficial from, again, I say my perspective on the academic side is that early engagement with regulators, it's very critical because it helps to create proper value proposition that the technology can get adopted by stakeholders outside their own research lab. Why would someone in industry, a nonprofit, would adopt a new lab? Because they see a value in it. And that value comes when the innovator is in contact with the regulators, so they actually engage and discuss it, and it helps with the model development. And I think that's very critical. And ultimately, I think another thing which was kind of showcased in the NIH talks about the Office of Directors, collaboration, complementary approaches. This is not a one-person show. This is like it takes a lot of people to be involved. And I think the more we push towards collaboration, and it's more like a top-bottom approach rather than people expected to be organically from bottom up to collaborate, which happens, sometimes doesn't. But if there's more leadership supporting the collaborative approaches, I think more innovation is going to happen. That's going to be a fruit coming out of it. And that's my 2 cents here. Yeah. Thank you.
Great. So we're going to open it up to the other SACATA members. If there's no clarifying comments, which I'm assuming there's not because there's no hands up. Was that a clarifying comment, Ellen? Okay. I am going to take the chair's prerogative. I have comments, so I'm going to go first. So. But keep putting your hands up and we'll open it up. So the OECD GD34 update, I just want to emphasize it's such an important activity, especially for industry who are going to be the ones that utilize the method for regulatory application. And I want to underscore the importance of ensuring inclusion for the ICVAM member voices. I know that we have a fantastic lead in Charles, but just to re-emphasize that there is a lot of different voices that need to be incorporated when we're getting this update put forth, so that we can have maximum alignment with the US as possible, global alignment. I'm going to switch around a couple of topics a little bit, but in the US, we don't have NAMs accepted by all agencies to replace the acute toxicity 6-pack. I know that I've said this before, but Even though the working group is complete, I know it doesn't go away, but I do encourage the ICFAM to continue the collaboration through regulatory acceptance, and try to facilitate that as much as possible. I have to mention the importance of making sure that mixtures and formulated products are considered and not just the single chemicals, and that specific work should be continued for, as an example, acute inhalation, for instance, the use of GHS mixtures equations by all member agencies, and as well as expanding skin sensitization into the IATA into mixtures in that application. It's fantastic, really fantastic to see the amount of cross-functional collaboration. I know this has been mentioned, but really want to emphasize the importance of this and encourage its continuation for all current and future projects. The case study presented by Amy was a great example of this, but there's also been many recent publications where you can, really see that cross-functional collaboration. And to underscore what was just said as well about including academia too, I think that's great. I do want to emphasize that regulators don't need to be the ones to drive and fund these efforts, although support and inclusion obviously is needed, like was just mentioned. We've got so many examples recently with projects being driven by non-regulatory entities, and especially with resource constraints that are typically prevalent in the regulatory sector. I think it would be even more important as we move forward to make sure that we're not just leaving it all up to the regulatory agencies to drive. And something also that was mentioned earlier, clear communication focusing on NAM's ability to protect human health instead of the obvious additional benefit of not testing on animals, having the main point of discussion being geared towards the ability of advancement of the technology to better protect human or animal health. This is going to aid in the acceptance and adoption of NAMs by all communities, even those that have historically been opposed to this space. So can't emphasize enough, we really, really need to have clear communication that emphasizes the benefits, not just the addition of saving animals. And I think this is last comment. Also to reiterate something that Sue mentioned regarding running side-by-side animal testing. I have lots of experience validating test methods against animal data, and it is possible to completely do that without additional animal data, right? So there should be no reason to conduct new animal testing for this purpose. Leveraging the historical data banks that many of us have, again, collaboration, if that's not something that the group that is currently working on these activities has, I'm sure that does exist. And then running the same compounds or formulations that were previously tested, that's a great way to tackle this. I cannot emphasize enough that really should not be any need to conduct new animal testing for validation purposes. Okay, so I'm going to go in the order on my screen, so sorry if this is not the order that was. Yes, sorry. Go ahead, Nicole.
Sorry, just to very quickly respond to one of the points that Dr. Page brought up, because it's been brought up a couple times today, on increasing ICFAM agency representation and participation in OECD work groups. Just to let the committee know that this is something that we have really ramped up over the last few months, particularly this year. We had a dedicated ICFAM teleconference where Charles and I went through with the OECD Secretariat and revisited the U.S. membership on all of the different OECD expert groups, sent that out to all of our ICFAM principal agency representatives and all of the members of ICFAM work groups, and asked them to disseminate that or review it with their specific agency leadership to ensure that the subject matter expertise represented on the OECD expert groups was current and appropriate, and people who had retired or otherwise moved on were no longer on the list. And there was a lot of refreshing that was done with those expert group memberships. So just to let you know that we are cognizant of that, and we've been very proactive about that recently.
That's really great to hear, because I think that is very important as we attempt to move forward in that global way. Okay, Ellen, go ahead.
Yeah, so great presentation on this session. I did want to make a few comments about Dr. Kovach's presentation on the OECD update plan. And I really appreciate that they understand the need to handle NAMS components separately and support the concept of technical validation. But it's not really technical validation, it's characterization. Because there's, and I think what we're missing is standards on performance, especially in vitro and in biochemical. What are they calling it? Chemical NAMS components to define the variability and uncertainties of individual assays. And I think there's an opportunity for us to, or for the group to figure out some standard metrics that could be used. And from a practical perspective on the workflow, I suggest that some of this prerequisite work in the readiness category, that's where you could get the performance metrics on a very small number of reference chemicals. Practical issue. So in place of, having to do ring trials, if we had an independent group who could prepare the reference materials blinded and then send it out to the different places, you could ensure independent and, you get data on the reproducibility of these assays. And many of these, especially the complex systems, CIDMs, aren't really easily portable, some of them. And so that would get around that issue as well. And maybe just the last comment, I was actually on the Methods Developer Conference, and there are benefits. It was great. There are benefits outside of what you even could imagine. So I presented on an early idea for an assay panel that we put away. We hadn't really thought about the application. So just having to put that together and get for the conference, it has inspired us to actually now go out for grants and sort of get those that assay platform, into sort of development as a real NAMS with all the requisite data that you would need. So just the spark of having those meetings, and method developers are so anxious to get in front of regulators. So I think that having those conferences, those groups is really a great advance this year.
Great. Dr. Nunez?
Sure. So my questions and comments will be for Dr. Rinke on the Method Developer Forum. I just first want to say it was amazing to hear all of the great progress being involved with this group from year to year. This is my second year. To actually see some of these things that we discussed last year come to fruition has been awesome. My first comment is it was really nice to hear you talk about the regulatory versus kind of pre-regulatory space when it comes to a lot of these assays. I think there's some things that I'll comment on here a little bit later of, things that we're missing where we could potentially have some impact here, especially thinking in terms of industry here. The other piece that I think is worth noting is the way that I like to think about this is in terms of the developers and then also the end user. So the developers can develop, but it doesn't always mean that it's going to be useful to the end user. And in this case, the end user could be pharma companies, agro, product, whatever. It doesn't really matter. At the end of the day, we're all kind of doing similar things where you start off with a bunch of molecules and you're trying to figure out, how to get the one that you're going to submit for regulatory type assays. So there's a humongous piece there that industry, a lot of companies already have their own kind of proprietary assays that they're using in their flow schemes that might be amenable to other groups. So I think it's worthwhile trying to reach out and kind of kick the proverbial can just to make sure that you're not missing anything that's already out there, not having to reinvent the wheel. So I think when I look at the slide for the future plans, it was amazing. You pretty much covered everything that I would think to put into this. So it was well thought out. It is a humongous amount of work. But I think that if you do it the right way, you should be able to get some really useful information that will not only impact the regulatory space, but also the pre-regulatory space. So I'll use your next meeting for cardiotox or the plans for that as an example. Well, yes, there's a bunch of regulatory studies that are required as part of the safety farm battery. Those are all very well known, but most companies are doing an entire other part pre-regulatory to help do kind of identification of their best molecules. So I think for something like this, it would be great not only to get some of the government agencies that you have listed here in your steering committee, but also try to reach out to members of the Safety Farm Society or to members, key leaders in all the different companies to kind of get a feel for what would be useful in that kind of space. So I think that pretty much covers all of the things that I brought about. But again, great work. I think there's a lot of room for collaboration, and I really like kind of the concept of this kind of method development effort. So.
Lily, go ahead.
Figure out which microphone. Yeah, thank you. I just wanted to mention two things. The Method Developer Forum is genius. I mean, it really, I think, even that model and using it in other places has so much opportunity for application. So, and just seeing the, having people come, this is the need, and seeing the variety of responses and the different answers that came forward was pretty impressive. So great work. I also did want to give a shout out to Amy's work. I felt like maybe it was a typo because it looked like you did the search on July 15th. Like that's 2 months. Like that's really impressive. Yeah. But just saying, like taking all that data, getting it to where she got it to present it here is spectacular. And also, I just see the applications, really widespread applications for that. So I just wanted to bring that up. I don't want to take a lot of time because I know you're trying to cruise through things, but that timeline was really spectacular. Nice job.
Yeah, I mean, collaboration, right? And then also re-emphasizing what I said about not necessarily waiting for the regulators to have to drive it. I think it's, that leveraging that partnership, it's really helpful because we all know that if we wait, sometimes it takes a lot longer if we're having to wait for regulatory resources. So collaboration is good. Misty?
Thanks. Great presentations. I just want to make a quick suggestion. I have a lot to say about the users and the method developers, but I think I'll just save that for later with respect to time. But as it relates to the method developers, one suggestion is to maybe take a look or talk to someone who did the HESI SIPA project around cardiotox. There's one vertical in there on iPSC technology and a lot of in vitro models and cell providers, and I think there's probably a lot of lessons learned from that maybe talking to Sterl, I think it's her name. But I'm happy to connect if you don't know them. But I think you can learn a lot to sort of move ahead on the curve.
We do have time if you want to. All right.
Why is it not changing?
Okay. I feel like we had a lot of time, but no, I know. I feel like we have time, but okay. Okay, so we'll move on to the next discussion topic, the NAMS pipeline for future directions. And Dr. Price, if you want to go ahead and kick us off.
Yeah, I'm happy to start us off here for the discussion on 2B. First of all, I'll just do what we've said a lot today, which is to say that it's always amazing to see how much progress is made within this group, how fast things move, how much you kind of see some of the things that were talked about last year get implemented, movement, and I think it really speaks strongly to the leadership and the team in terms of what they're doing. So I'll just focus in on a few things here at the end of the day. So one of the things that really strikes me when I look at this is how much overlap or how much connection there is between lots of things that we do in, say, personalized medicine or personalized health, personalized nutrition even to some degree, and the flip side of toxicology, right? Looking for molecules that are beneficial, looking for molecules that are anti-beneficial. It's pretty much kind of two sides of the same coin. And so, it does strike me that so many of the elements that we dive into in terms of digital twins, which was brought up in this session, N-of-1 analysis in terms of understanding what's happening in one individual's body with multi-omics analyses. I'll get into some of these in a little more detail. Just that there's a lot of connections, and it was great to see the big consortia that was talking with the Common Fund and so forth that was pulling in, so many. I might be mixing talks here, 18 different agencies, but I feel like that was Nicole that said 18 agencies. So maybe she beat you, I don't know, or she's behind. But anyway, bringing in lots of different groups. I do look at some of the other big initiatives that are going on. ARPA-H, for example, or DARPA are engaged in a lot of really big computational modeling initiatives now. There's probably possibilities for connections between NIH and some of those groups as they come up. Likely important to coordinate and try to take advantage of those things. Was brought up the importance of combinatorial NAMs, and I think that is incredibly important because there's so many interactions that are possible. It's obviously impossible to screen all combinations, just given combinatorics. So the continuing improvement in computational strategies using in silico methods to guide that, I think, is key. And I am very interested, looking forward at, how you can really incorporate screening, AI, mechanism understandings, and so forth to really probe that combinatorial space and really automate it in lots of different ways, because there should be abilities to put in line AI, robotics. I think mechanism is important for the reason that you can't just brute force all these things. So, to the degree that we can try to inform combinations and so forth, but that feels to me like an area where capabilities in screening could be probably exponentially enhanced by figuring out how to do that in really good ways. The recommendations that were given from the working group, the A1s, I thought were really good. I agreed with all of them, and trying to find the ability to get high quality interoperable data sets, which was brought up, and creating alliances, to manage, share these kind of high quality NAMs, I think is really important. There was a lot of discussion about engagement. This is a huge element, obviously, about how to really drive these forward. And there's so much that kind of has to be done in terms of what are the really big drivers that would make people identify these. There was a lot of discussion around making it as easy to use as possible. I know there's initiatives that are ongoing to kind of summarize a lot of that information so people have a one-stop shop to go ask what alternative methods exist. Workshops was listed some ways for ideas to engage. I do really like the notion of trying to do something like that that's very practical. Maybe if it's oriented towards even towards an industry event and so forth, like you can come in and bring the product you want, the molecule you want, whatever it is, and go through with experts in terms of how you could think about that regulatory pathway. I still think one of the pieces that's missing, unless I didn't hear it, is I still like the idea of identifying like one area where NAMs can be better, cheaper, faster, like the preferred method to an existing path, such that you could get widespread adoption in one particular area that kind of becomes an example of like just strong success stories that then go to other groups. You can certainly in the comments back to me say that was obviously shown and you missed it. Just tell me when, because I feel like it would be nice if we had a little bit more structured in one of those areas, right? Which is how good are the animal models at transitioning compared to the NAMs that we have it better, that is cheaper, et cetera. Because I feel like it's kind of a hard bar because the other path is much older, right? Better resource for a long period of time, and so we're sort of the upstarts. But I still feel like that's one of the things that would make a big difference. The complementary program I thought was super interesting. Very interested to hear about some of the centers that are being set up. I think that sounds like a great program. Maybe I'll go for one. I don't know. We'll see. So other people may as well. But those are, I think, exciting to pull people together. Scientific needs that were brought up. I did really like this aligns with a lot of what I think about a lot, but around personalized health. And I do think that there's more and more of these programs. Will be running a number of these, a number of them emerging that are trying to generate like really dense multi-omic data from longitudinally from individuals. It's been done on thousands of people now. I think those kinds of efforts are really big opportunities for the toxicology space. Again, on this two sides of the same coin type of idea, where we can really try to get a sense in those big longitudinal studies of kind of the effects that a lot of this toxicity is observed when we find it in people's blood. There's also some really interesting connections, I think, with genetics. One study we did a few years ago was quite interesting, looking at people that were asymptomatic but just had high genetic risks for ALS, for example. And it turned out that they had unusually high levels of heavy metals. At least it seemed they had high levels of heavy metal in their blood. But not because of, as far as we knew, because of environmental exposure, but because the polygenic risk score for ALS seems to be associated with a worse ability to clear heavy metals, for example. And that was just incidental finding that we, came across in our studies. But I wonder how much of that is out there. Let's see, coordination centers. Yeah, I think with that, I'll just say then also the foundation of the NIH, obviously those, building bridges to breakthroughs, the connections of with public-private partnerships. I think those are incredibly important. And especially as I was mentioning, this area of precision health, I think is a huge conduit because there's lots of these private companies that are trying to develop more and more of this. And so there's just going to be so much more data opening up what's going on out there, and could be just really great interfaces with the great work that's happening here. With that, I will end and turn it over.
I can't really resist. Nathan's like, tell me I'm wrong, that there's not a success story.
and I don't mean to say there are success. Like I said, I'm very serious. Like this is an amazing group. It's going quickly, but I still feel like that one story where the whole industry like shifts away.
Yeah, I think the closest thing that we have
that we own the most.
Yeah, the closest thing that we have is probably skin sensitization, but more sort of the whole umbrella of acute topical toxicities. So skin irritation and corrosion, eye irritation and corrosion, and skin sensitization are three endpoints where we have very conclusively demonstrated that human biology-based approaches are better, faster, the cheaper part is arguable at the moment, but as it becomes more widespread and more commercially available, then we'll get to cheaper as well. Yeah, thanks.
Thank you. Patricia Silveira, second member. So, in terms of speaking about some of these, some of these strong questions. So, as an academic, I want to emphasize the importance of training and dissemination of all of these NAMs and these applications, particularly incentivizing people to attend workshops and learn more and communicate them, and especially those repositories, those protocols, and like the standardization that is much needed, especially in the academic side. We often go with what is going to be accepted by the reviewers, accepted by the community. So just having this confidence will accelerate the implementation. And I know that in these meetings and these approaches that you conduct, you have these multi-stakeholder communication, and there's a lot of innovation that is being incentivized. But I think in academia we're a little bit more laid back, and we need more validation and more education. So along the term, I wonder if some of this funding that is being put forward to incentivize use of these NAMs has any intent to support educational activities, for example, curriculum development, or integrating this into like toxicology programs and training programs, so that the next generation of researchers that will certainly be using these technologies on a day-to-day basis, hopefully, could get to learn as these things are being developed and implemented. And the same for goes to how the statistical approaches and the methodologies that are used to ensure that this is rigorous. And how do we define rigor in this space would be beneficial for us educators to help accelerate this. In terms of the second thing that I would say would be a priority, I really like the idea of these providing resources, infrastructure, and funding for collaborations across multidisciplinary teams. But also, I think it's important to incorporate metrics and train the reviewers and provide some sort of framework for the review bodies when you're not just responding to a specific RFA. So, for example, if I want to use NAM in like my regular research program, maybe I will encounter reviewers that will say, well, this is not good, this is not validated. So I wonder if all this, if we can integrate it beyond just this new thing that is specific for these more selective group of people. And I'm not trying to be critical of you, but I feel like I only learn about these things when I come here. And then I go back to my academic center, and there's very, very few people that would adopt this because it's not that much disseminated. Yeah, more of that. And then I really, when we talk about the second topic, the engagement between complementary and ICBAM, I really commend you for this intersector approach and having those listening sessions. And there's a question about how to measure success. I wonder if, how do you set your goals for progress? It will be more effective if you have a baseline where you're going to compare to. It's hard for me to talk about it because it's not really my area, but I think, if you want to define more people publishing with it or more people adopting it, or what is the metric, is something that perhaps will help you establish the success of the program. There was something in one of the presentations that talk about, all these communities and all these people that are being brought in to comments, and I'm not really clear on how the communities are selected, like who is your audience that you're trying to target, and maybe considering expanding that or see who thinking outside of the box maybe will help get a new perspective. And then for the last item in the integration, in the validation network, I really enjoy listening at how NAMs are being incorporated and how the data is being shared across people and integrating everybody's feedback. I think you should continue to ensure disseminating where the lessons learned, what works, and also making sure that you, in these outcomes, the physiological variables, like the ones I mentioned earlier, and like the changes across the lifespan, what Nathan was talking about, the things that happen during life and then later on are crucial for the development of sustained outcomes, are disseminated, are integrated, and that the information is properly shared so that these can be implemented faster. Right, that's it. Thank you.
Great. If there's no clarifying questions, we'll move on to Dr. Baines.
So the problem with going last is everybody says everything you want to say. However, I do have a lot to say still. So first of all, I just wanted to commend everybody who's been involved with all these efforts. Again, when I think about, I've been on Cicadum for a couple of years now, and to see the growth and all the things that have happened, it's really cool. It's really cool and amazing to see the progression that's happened up to this present moment. So again, I turn my video on too. Yes, I can do that. I can do that too. Yes, I can turn my video on. Yeah. Yeah, you good? Yeah. So video on and mic is on. Yeah, so I'm good. I think I got it. But yeah, so I just want to commend everybody who's been involved with this, with this space, because it's been like a tremendous growth over the past few years since I've been on this committee. So again, commend everybody, working groups, ICM, everybody. So just a couple of, comments. I'll start off by when I first came on campus, NIH campus, talking to my colleague Nathan, catching up since I hadn't seen him in a year. We talked about, what we've been up to. And, again, I studied cancer. We talked about cancer and talked about how, we still don't do a great job treating cancer, in terms of metastasis. I studied pancreatic cancer, which is, horrible in itself. And then, we brought up glioblastoma, which is another horrible cancer. So again, at the end of the day, we still don't do a good job when it comes with dealing with metastasis and drug resistance, being a pharmacologist, toxicologist. And, we use tons of animal models for those studies, and we still, like I said, don't do a great job. So I think, again, just a great example of how we still need to bring everything, all resources to the table, including NAMs, to help fight these different types of diseases, and so I just want to bring that up. So again, I applaud everybody who's working in this space. Also, I actually had the opportunity to be on this working group that for the NIH director dealing with NAMs, and I was totally honored to be on that working group. And I definitely, it was definitely a team effort and a lot of work. So I applaud all my colleagues on that committee with me who came up with these recommendations. And so as it relates to the recommendations, all of them are important. So, it asks which recommendation will you prioritize. I'm like, I don't want to get in trouble with anybody with these recommendations, but, all of them are important. So I want to be clear, all of them are important. All of them required a lot of work and conversations with different stakeholders. But there's just a few that I wanted to just bring up that I think may be worth prioritizing. So one is recommendation number 2, establish resources, infrastructure, and collaborations to promote the use of interoperable, reliable, and well-curated, high-quality data sets produced from research using them. So again, totally important. But if you don't have the infrastructure and the resources and collaborations, none of this is going to happen. So that's definitely a recommendation that I would prioritize. and trying to work on. Another recommendation, recommendation 3, promote effective dissemination and interconnection of NAMS technology. We had a session at NIH, actually it was kind of like a year ago here, in reference to these recommendations, and I moderated a session on basically equitably deploying robust and reliable NAMS into practice. So I just wanted to make sure we keep that in mind, that we need to make sure that when we disseminate this technology, that we're making sure we're keeping everybody in mind. I immediately think about the issue with cancer. One of the model systems that we use are cancer cell lines, and many of those cell lines are not diverse. They're very homogeneous cell lines. And the problem is, if you're trying to answer questions about cancer in different groups, will a very homogeneous cell line tackle that? And unfortunately, probably not. And so, I just want to encourage us, as we are still on the front end of NEMS and really pushing this field forward, that we keep in mind that we make sure we do all we can to make sure it's an equitable dissemination of this NAM technology. So we're not looking back on the back end like with cell lines. Oh, now we need to collect cell lines from other groups of populations. So just want to just make us aware of that. And then the last recommendation, again, there was I think 7 recommendations. All of them are important. I want to be clear. But I guess the third recommendation I would, prioritize is recommendation 5, facilitate multidisciplinary teams with expertise across technologies and the life cycle of NEMS development and use. Basically everybody needs to be at the table. There's no special, I'm a pharmacologist, biochemist. It doesn't matter. We need everybody, statisticians, epidemiologists, public health workers. We need everybody at the table on these different teams. So we talk about all these different teams, all these different working groups. We need to make sure they are inhabited with all disciplines, chemists, biologists, physicists, mathematicians. We need to make sure everybody's at the table, because again, I think when everybody's at the table, we make better decisions and we tend not to leave anybody out. And so just applaud. So again, I just encourage us to keep that in mind. So again, all 7 recommendations that the committee came up with are important. Those are just 3 that I think definitely need some attention brought to it. I guess as it relates to the complementary program, again, great program. I think it's so cool that's coming to fruition. I can see a lot of benefits from that program in terms of complementing studies with animals, or sometimes we place them all together. So again, I just think that's I think just moving in the right direction. I guess I would just say a few things about that. One, I think it's important to make sure that I know there's funding that's provided, which is, I think, training, these competitive grants. I think they're great. But we just don't want the same universities, same institutions, same large companies getting these grants. We need to make sure that there's, and I want to be clear, I don't want to exclude anybody from getting these grants, but we just need to make sure that there's opportunities for smaller universities, smaller companies to also access these grants. We just don't want it to be, again, I want to be clear, there's no shade here against large Ivy League schools or large research institutions. We need everybody at the table. I just want to be clear. We need everybody at the table. However, we want to make sure that there's opportunities for, smaller companies, smaller universities, medical schools, different medical schools, smaller medical schools. And we just want to make sure that there's opportunities for different entities to be able to also compete for these awards. Because if not, again, we may still get into this space of a very homogeneous thought process. And so, I just want to encourage us to keep that in mind, that we just, again, at the end of the day, it's about, diversity, inclusion, equity in all spaces, when it comes to opportunities, to come up with some really good ideas. And these ideas are not just, isolated to like a certain small group. It's, everybody can bring something to the table. So I just want to just encourage that as we move forward with providing grants and opportunities for this. And I think, ICBAM can really be involved in helping to make sure that, again, going back to what my colleague said, Patricia, that we need to make sure that there's training and communication. I read some of the public I read some of the public communications, and there was concerns about transparency. I think I read about with the FDA and stuff. Transparency in terms of NAM's NAM's work being done and everything. And again, I don't know much information about that specific situation, but I think at the end of the day, we need to do, there's always room for improvement in making sure we communicate, what we're trying to do in terms of NAMs and make sure we are as transparent as possible, as we move forward. And that information is not just for scientific colleagues, not just for other academicians and folks in regulatory, it's for everybody, even for the public. I mean, I think, sometimes I hear science doesn't seem to be really that important until it's on the radio or in some magazine or a newspaper or social media. Well, I think, if we try to really, get the word about NEMS and the importance of NEMS, I think we need to make sure we get NEMS in that space too. Yes, it's good to have the publications. I think it's awesome you have over 200 some publications. That's amazing, But again, at the end of the day, who's going to read those publications? And yes, scientists will read them, maybe, but really we want to make sure the public's in this space too, because the public is going to be, helping to make decisions on some of these policies. So I think it's important to make sure. So I think ICVAM can be helping out with making sure that we are being that the complementary program is being as transparent as possible. We're working together, and that we're hosting workshops and educational sessions, for all stakeholders. And those stakeholders need to include students because again, we all are at various levels of maturity. And so we need people to replace us. And we want those folks to have the same, we want those folks to have a very strong foundation on NEMS and the importance of NEMS. Again, a lot of us came up during a time where that wasn't the focus. But, nowadays we realize that, we can use all tools and resources to help, guide our science and our research. And so I think it's just important to make sure that we do, do our due diligence in educating the public and all stakeholders, including students, grad students, even undergrad students coming along. So, so all the work that we're doing now won't, fade away, once, we retire or whatever. So I just wanted to make that case. So that's one of the things I think we can do to help make sure that this knowledge gets transported down to future generations, which is going to be important moving forward. And then I guess, again, the last thing I guess I'll say is, especially as it relates to the last question about validation and, initiative. So I think it's important to make sure, again, that we, remember what we're doing, that we're trying to improve biomedical research. We're trying to solve some of these problems, these health problems. And we need to remember all groups that are involved. Again, when it comes to health disparities, when it comes to, these different diseases, we need to make sure that we're keeping in mind who we're trying to benefit and do all we can to make sure that they are involved in conversations on the front end. These partnerships need to happen on the front end. A lot of times, you have these partnerships, and then after these partnerships form, okay, we forgot to ask this group, and then you ask them on the back end, and that leads to distrust and all those interesting issues. And so, again, I just encourage us that NEM is like moving full speed ahead. All these working groups, all these partnerships that's forming, that we do our due diligence to make sure that we make sure those groups, those partnerships include everybody on the front end, so we're not having to try to fix things on the back end and not actually benefiting everybody and not just certain groups of people. So just wanted to highlight that. But again, I just want to applaud all those that are in this space that have really pushed NEMS to a great space over these past few years. So I just wanted to thank you for that. I'll stop there.
Fantastic. Okay. I am not seeing any hands up right now, but we will open it up to the rest of the TCATM members at this time. If there is no additional comments, doesn't look like there are. Okay. Well, with that, then I'm going to spend 30 minutes to summarize. No, I'm joking. Okay, so just making sure everyone's awake. Okay, so with that, we will adjourn day one. I do want to remind everyone that there is a slightly earlier start time tomorrow. We do want to start 9:30 a.m. promptly. So see you all tomorrow. Thank you.